Hello, everyone. I'm Professor Hyukjin Lee from Ewha Women's University. I'll be a moderator of a symposium. I'm very happy to participate this event with former colleagues. Now I'd like to introduce Professor Do-Sung Lee. Professor Lee is the Vice President of Korea Academy of Science and Technology. He is going to give us a welcome remark. Let's welcome.
Good morning, good afternoon from US. Distinguished guests from home and abroad, welcome to the 48th KAST Online International Symposium. I'm Do-Sung Lee, the Vice President of the External Relations of the Korean Academy of Science and Technology, and also a professor of Sungkyunkwan University. On behalf of Korean Academy of Science and Technology, it is a great honor and pleasure for me to welcome you to the 48th KAST Online International Symposium entitled Recent Advances in the Development of mRNA Therapeutics and Their Delivery Systems. My special gratitude goes to the speakers for making this symposium possible. I'm delighted that KAST can relish a role that provides these scholars to exchange scientific achievement. Also, I'd like to extend my sincere gratitude and appreciation to Professor Hyukjin Lee, the organizer of this symposium. It is a tough time for all of us due to the COVID outbreak. However, there is always opportunity for new technologies to shine at the right time and point. Since the mRNA COVID-19 vaccines development, mRNA technology has been in the spotlight, and scientists across the world are eager to harness it to make a variety of drugs and therapy and vaccines. I believe in vitro transcripted mRNA and lipid nanoparticle delivery systems are two most successful technologies standing out for the COVID vaccine development. Due to the huge success of COVID mRNA vaccine, there have been tremendous attention on the development of future mRNA therapeutics and delivery systems. KAST has noticed this growing interest and able to organize online international symposium open for the public. Today, we invited 7 outstanding speakers from US and Korea to overview the recent advances of this development. I'm very proud of having today's speakers and looking forward to hearing their talk. I hope that you will enjoy a successful meeting from the viewpoint of not just knowledge exchange, but also mutual collaboration. Thank you very much.
Okay. Now I'd like to introduce Professor Yu-Kyoung. Professor Oh is the chair of College of Pharmacy at Seoul National University. She will be chairing the first session of the symposium. Please welcome Professor Oh.
Thank you, Professor Hyukjin Lee. I am very glad to chair the first session. So today I'd like to introduce the first speaker, Professor Dan Anderson. He is the professor at MIT, and most of you knows he is a global expert in the delivery system of various RNA, such as siRNA and mRNA and sgRNA. He is the founder of the many venture companies and biotech companies, and he founded ORNA and CRISPR Therapeutics. Please please join me to welcome Professor Dan Anderson. Dan?
Terrific, thank you. Let me share my slides. Great. Well, thank you again for the opportunity to speak. It's my real pleasure. Can you all see my slides now?
Yes.
Yes,
Yes.
okay, thank you. So what I'd like to do is really talk about our work on nucleic acid delivery systems for RNA therapy and also genome editing. But first, perhaps give a little bit of a background on the field and one of the, a little bit of perspective on why I'm so excited about it. I think there's really no question today that the intracellular delivery of nucleic acids will revolutionize medicine. And, this is really not a biological challenge. There's no question that these molecules can encode therapeutic proteins and regulate different important genetic pathways in cells. The real question is, how do we deliver them inside of cells? And how do we do that in a safe and effective manner? And, what's interesting today is that, at least in Massachusetts now, most people have gotten injected with mRNA lipid nanoparticles. And these have really changed the life experience of those of us out here in Massachusetts, and we expect more broadly in the entire world. What's perhaps less well known is that the chemistry and technology behind the COVID mRNA vaccine really grew from efforts to deliver RNA to the liver, and in fact, small RNAs for gene silencing. And so just as a refresher for those of you that haven't thought about RNA interference before, RNA interference is a fundamental pathway whereby double-stranded RNA is cleaved into small fragments. These small fragments of around 20 to 22 bases can then interact with an endogenous enzyme system, the RISC pathway, to form a complex whereby one of those two strands is loaded into the complex and will seek out the complementary messenger RNA to destroy it. And so this is a catalytic nuclease that is basically silencing a specific gene depending on sequence. And it wasn't long after the discovery of this pathway that scientists and doctors and even investors, of course, realized that if we could just deliver small RNAs inside of cells, we have the potential to silence essentially any gene that we want. And so my colleague, Phil Sharp, coined the term modular pharmacology for this vision of creating drugs that just by changing the sequence of the RNA, you may be able to silence any therapeutic gene target that you want. Oops. So again, the challenge was not biological. It was really a delivery challenge. How do we deliver these complexes inside of cells? And if we envision this process, it's actually a multi-step process with many different challenges. First, we need to condense the RNA into nanoparticles. And then once injected into the body, these nanoparticles need to travel through the bloodstream to reach our tissue target. They need to leave the bloodstream and interact with our specific cellular targets, inducing entry, and this typically occurs through endocytosis. Once endocytosed, they're trapped in this endosome or cellular stomach, and they need to escape that endosome. And then finally, of course, the nanoparticle needs to decomplex to release the RNA so that it can interact with the cellular machinery and have its therapeutic effect. And so, it seems like an impossibly challenging problem, but there's been progress. And where we first saw progress with these types of particles were in organs that were natural destinations for injected nanoparticles. There are many organs in our bodies that are constantly filtering the blood, organs like the liver, the spleen, the kidneys, to some extent the bone marrow. And it turns out that these are, of course, natural destinations to injected nanoparticles. That said, despite the fact that many nanoparticles will accumulate in these tissues, that's not the same thing as having intracellular delivery of a payload inside of the cells in these tissues. And so the real challenge was not just getting to the organ of interest, but also entering those cells in a safe and effective manner. And unfortunately, RNA and other nucleic acids are simply not great drugs. On a drug scale, they're relatively large molecules. They're highly charged. They do not cross cellular membranes. They're prone to nuclease degradation and can be degraded if they're not protected. And they can even induce immune responses through TLR pathways and pattern recognition receptors. And these can result in toxicity and also dysfunction of the payload. That said, as I mentioned, we've all seen progress. And in my mind, these are really the three keys to making nucleic acid drugs. The first is in proper sequence selection. So a lot has been learned about how you can identify the optimal sequence for an siRNA drug or also for a messenger RNA. So in the case of siRNA, we're looking for small RNAs that are very potent, but also specific in that they do not silence genes we're not trying to target. So there are now algorithms that allow us to identify these sequences without having to make thousands of different molecules. Chemical modification. So a huge amount has been learned over the last few decades about how we can chemically modify RNA to give it drug-like properties. So this includes changing the bases, the backbone, and thereby introducing resistance to nucleases. We can lower these non-productive immune responses, and today we can even, to some extent, add ligands that can allow the RNA to deliver itself, at least in the case of small RNAs. However, the thing that I've sort of spent most of my time thinking about is encapsulation. So building nanoparticles that can actually act as trafficking agents to deliver our RNA payloads to the cells. And this is particularly important in the case of messenger RNA and other long forms of nucleic acids where we can't use all of these chemical tools. So a fundamental question, if we're going to build a nanoparticle to deliver RNA, is what materials might we use? And this is a sort of canonical composition of an RNA lipid nanoparticle. So I think if you look broadly across the field and at what companies are doing, you will often see that these lipid nanoparticles have four main components. First and foremost, they have an ionizable lipid. So this is an amine-containing lipid or lipid-like molecule that can both interact with the phosphates of the RNA to condense and self-assemble into nanoparticles, but can also interact with the lipids of the cell and the endosome to facilitate cellular entry. These lipid nanoparticles often typically contain polyethylene glycol lipids, as I've shown in the bottom, whose role is to facilitate colloidal stability and circulation of these nanoparticles. And they will often have other helper lipids, other lipids like DSPC, phospholipids, cholesterol, and these play a variety of roles, including stability and also affecting biodistribution. So through very simple controlled mixing, these components along with RNA can be complexed into nanoparticles, typically on the order of around 80 nanometers or smaller. And it turns out that one of the key components in all of these lipid nanoparticles really is the amine-containing lipid. And so when we first started thinking about making these, we were, of course, naturally inspired by all of the great work that had been done before us with antisense and DNA delivery, dating way back to the 1980s. However, when we looked at the amine-containing lipids that had been available at the time, we noticed that there was not a huge amount of chemical diversity. And so, for example, I've drawn two structures. DOTMA is a lipid in the upper left corner. This is from the 1980s. DLN-DMA was an important early lipid for siRNA delivery published in Nature by a company called Protiva, I believe around 2006. And when you look at the structures of these two amine-containing lipids, they're really only different by a few carbons. And so that's not to take away from the importance of this lipid DLN-DMA, but rather just to indicate that perhaps the structural diversity when we first started was not as broad as it could be. And so the first question really was a chemistry question. How do we go about making as many of these compounds as we need to? And so we developed a variety of chemical procedures to rapidly make lipids and lipid-like materials that's amenable to parallel chemistry and combinatorial development. So here I've shown one example chemistry, which is a Michael type addition between an amine and an acrylate. And this is a very simple chemical reaction where just by adding heat in a stir bar, we can rapidly develop literally thousands of different molecules. And we've gone on using this and other chemistries to make libraries of amine-containing lipids and evaluate their performance as RNA delivery molecules in a variety of settings. So an important first example of an RNA lipid nanoparticle is siRNA therapy for the treatment of a disease called transthyretin-mediated amyloidosis. And so transthyretin-mediated amyloidosis, or TTR, is a disease caused by misfolding of transthyretin, which is primarily expressed in the liver. And because of this misfolding, amyloids can form, and they can then, from the liver, distribute to various tissues, thereby causing toxicity, both in particular to the heart, but also to nerves. And unfortunately, after symptom onset, life expectancy is short. And previously, really the only real treatment for bad cases of this was liver transplantation. However, it's possible to make a nanoparticle that includes an siRNA that is going to turn off this TTR gene. And so if we imagine a nanoparticle made with an siRNA and these 4 canonical lipid components, we can formulate that, inject a primate. This nanoparticle will travel through the bloodstream of the primate to reach the liver. And once it enters the liver, what we can see is we get dramatic and specific silencing of the TTR gene. So this is actually an old study. This work was first published in 2010. And so what you can see here is after a single injection, in 2 different studies in primates, we can get very dramatic silencing over 90%. And even at the lowest doses of siRNA, we're still seeing very good silencing of this gene target. And so one of the interesting features of this system was that we found a variety of lipids that could actually work in primates. So in our lab, actually, here are 3 structures of lipids that, when formulated with siRNA, could induce knockdown in primates in the liver. And other companies and labs have started to develop other lipids that can do this as well. Importantly, Alnylam took a lipid nanoparticle formulated with an siRNA targeted TTR into the clinic. And so this is an example of clinical data where patients with ATTR, this disease, are treated with this lipid nanoparticle. And after a single injection in these patients, what you can see is over 80% knockdown of the TTR gene. And through repeated administration in these patients, that silencing can be stably reduced 80%. And it turns out that this actually has a profound effect on the disease, and I'm not going to talk about that right now. But ultimately, this resulted in approval of the first RNA lipid nanoparticle in August of 2018. And this was an exciting moment for those of us that have been working in this field for a long time, because it really showed that these RNA lipid nanoparticles were not just powerful tools for inducing gene silencing in animal models, but actually could be approved as drugs and actually help patients. So an interesting feature of how these lipid nanoparticles target the liver is that there are natural processes whereby lipids are trafficked to the liver. So depending on where you are in the world, if you're going to eat breakfast or dinner and you eat fat, that fat will be absorbed in your gut, enter the bloodstream, and then will interact with a variety of factors in the blood, in particular, one of which is ApoE. And through a variety of processing steps, what happens is these dietary lipids are complexed into small nanoparticles on the order of around 100 nanometers called chylomicron remnants. And one of the jobs of our liver is to actually scavenge these dietary fats and process them. And so it turns out that with some of these lipid nanoparticles with RNA, what happens is once they're injected in the blood, they become associated with this endogenous ApoE protein. The nanoparticles travel through the blood to the liver, where they enter and interact with hepatocytes, and specifically with this ApoE receptor. This triggers endocytosis. And then the nanoparticles, once endocytosed, there's a shift in pH that causes a variety of changes in how these lipids interact with the membrane of the endosomal stomach, the cellular stomach, or the endosome, ultimately leading to release of RNA. This is an interesting slide I thought I'd show that just sort of shows the process that the Alnylam team, in collaboration with the University of British Columbia and a company called Alkena, and also our lab at MIT, together developed nanoparticles with increasing potency over time. And so what you can see here is that on the y-axis, we have knockdown of a target gene factor 7. And on the x-axis, on a log scale, we have the amount of RNA needed to get this biological effect in mice. And so the very first nanoparticles that we saw required very high doses in animals to get any silencing. However, over time, a series of nanoparticles were created by by all of us that dramatically improved the potency all the way down to this one, which is literally 5 orders of magnitude more potent than those very first lipid nanoparticles. And so I think there's 2 important points. First, this shows that by iterative, hardworking development, it's possible to create very potent delivery systems. First to the liver, as we've shown here, but I think it's going to be possible to do this to other organs. I also wanted to mention actually that when we published this paper in 2014, it turns out that 3 of the speakers here, Yizhou, Gaurav, and Dan Siegwart, are all authors on this paper. So it's a great pleasure to be able to give a talk with people that I've worked with over the years and all of this great work. One thing that I wanted to mention was that nanoparticles made out of lipids are not the only types of nanoparticles that can be used to deliver RNA. So it's also possible to use polymers. So here's an example of a polymeric, a recipe for a polymeric RNA nanoparticle. In this case, it's a combination of a biodegradable amine-containing polymer. In this case, it's a polybeta amino ester. And then optionally, we will add those helper lipids, the PEG lipids, the phospholipids, cholesterol. Sometimes these are added and helpful, and sometimes they're not. So I want to just give one example of a polymer-mediated delivery system. So we developed these small polymers that could actually facilitate delivery of RNA to the endothelium of lungs. And so in this experiment, we took 5 different siRNAs targeting 5 different gene targets, created our nanoparticle, inject a mouse, and then we go on to look at silencing of all 5 of these gene targets in animals. And what we saw was after a single injection, actually all 5 of these gene targets are silenced. So that's Ty1, Ty2, VEGFR2, VE, cadherin, and ICAM2. And as you decrease the total dose of that nanoparticle collection, you still maintain good silencing in animals. And so this is an exciting slide to me for 2 reasons. First, it shows that lipid nanoparticles are not limited to silencing genes in the liver. They can be used in other tissues, in this case, the endothelium. Second, it also shows the possibility to multiplex these nanoparticles to target multiple genes simultaneously. And so I work in a cancer center, and I work with biologists who are interested in complex pathways. And so we can envision building nanoparticles that can silence multiple pathways simultaneously, in this case, 5 genes, but we've even done experiments where we've been able to hit up to 20 in a single formulation. This has gone on to be shown to be effective in primates, and there are companies now working to advance this technology in the clinic. I want to switch gears now to messenger RNA, which is, of course, the focus of this symposium. So messenger RNA, of course, is part of the central dogma of molecular biology, as we're taught in the US in our biology classes. So DNA makes messenger RNA, makes proteins. And so there's no question that messenger RNA can make therapeutic proteins. As with siRNA, the question is delivery. How do we deliver this messenger RNA inside of cells? And so in my mind, I actually think of messenger RNA therapy as the other half of modular pharmacology. We turn genes off with siRNA, but we can turn genes on or introduce new genes with messenger RNA. When we think about the delivery challenges, I have already mentioned that small RNA is a difficult drug because of its chemical properties, its size, and susceptibility to enzyme systems in the body. Messenger RNA is even more difficult. It's extremely large. It has a limited set of chemical tools to allow for synthesis. And because it's single-stranded, it's even more susceptible to nucleases. That said, clearly, messenger RNA can be developed into a therapeutic. And so I thought I'd first give an early example from our lab where we took messenger RNA encoding human EPO. So EPO, or erythropoietin, is a hormone created in the kidney that will induce red blood cell production from the bone marrow. It's used to treat anemia. So in this case, we created a nanoparticle with a messenger RNA encoding human EPO. We inject a mouse, that nanoparticle goes to the liver, and now the liver becomes a factory of human EPO. And so after a single injection in these early experiments, we could get over 60,000-fold the physiological levels of human EPO in the blood. And in fact, since then, these numbers are now much higher. it's been demonstrated that you can make a range of different proteins, not just EPO, but essentially any gene that you encode in these messenger RNAs, including other therapeutic proteins and antibodies. And primate efficacy has been demonstrated, and now these types of nanoparticles are also in clinical development. An interesting feature of these systems is that it's also possible to build nanoparticles to travel to specific parts of the animal. So here's just one slide using a messenger RNA encoding firefly luciferase. And so what we see here in this experiment are two different nanoparticles, one that will deliver to the liver and the other that will deliver to the lung endothelium. And so what you can see when you look at the luminescence from the animals on the left, we have the lungs, and you'll see this first nanoparticle, which I've labeled PBAE because it's a polymeric nanoparticle, will give us good transfection of the lung, but essentially no transfection in the liver, no transfection in the liver, as shown here. And in contrast, the second nanoparticle will give us very little to no transfection in the lungs, but great transfection of the liver. So again, it's possible just by changing the chemistry of these nanoparticles to see trafficking of messenger RNA to different tissues. And I think that's a really increasingly interesting feature of these LNPs, that they will be useful in a range of different parts of the body. It's also possible to nebulize these nanoparticles. So you don't just need to inject them into your blood, or in the case of vaccines, into your muscle. They can be created as a mist, where you subject an aqueous solution to a vibrating mesh that makes small water droplets that can then be breathed in. And so here's an example where we took the same firefly messenger RNA in a lipid nanoparticle, and we have the rodents breathe that in. And what's interesting here is you get very good transfection of the lung epithelium, so the inner lining of the lung. And it's really focused to that tissue. When we look at the other tissues, kidney, liver, spleen, we see no transfection. So again, messenger RNA is not limited to the liver or limited to vaccines. It's going to be useful for a range of different tissues. And one sort of final point I wanted to make is the possibility of using messenger RNA to do genome editing. And so this is actually a problem that brought me to MIT back in 1999, was sort of the dream that you could create a nanoparticle that could actually fix your DNA while you're still using it. And so if we draw a cartoon, imagine if you have a liver disease. That's why I've drawn this liver in green. Can you inject a nanoparticle that could travel through the body, reach the liver, enter those cells, specifically cleave the DNA in a way that is therapeutic and ultimately results in a restoration of function? And so I think there's been some really exciting developments over the past few years to demonstrate the possibility of this approach. And so there are a really interesting growing list of different ways to do genome editing in people. I think for many of these, they rely on endogenous DNA repair pathways that we all have in our cells, and inducing events that will cause our own DNA repair pathways to edit our genomes in a way that's therapeutic. And I only want to give one example that includes the Cas9 enzyme system. So as I expect you know, the Nobel Prize was recently awarded for the discovery and use of Cas9 for genome editing. This is a bacterial enzyme that can be targeted to the genome through use of a guide RNA to make a specific double-stranded break. And I want to give just one example, which is using that Cas9 system to permanently turn a gene off. We can target it to a specific site through use of a guide RNA. It will create a double-stranded break. And then if it's rejoined through non-homologous end joining, it will be cut again. And this process will continue until small mutations occur that result in what's called an indel mutation. This causes frame shifts, which ultimately inactivate this gene. So to do this, we wanted to use messenger RNA encoding Cas9, but also wanted to deliver a guide RNA. And the question was, how do we also deliver the guide RNA? Well, it turns out that these same chemical modifications that people have been looking at for antisense are particularly useful in delivering of the guide RNA. And the question is not so much exactly what types of chemistries are useful, but precisely which bases on a very long guide RNA can we modify and retain function, and which bases should we not modify because they might limit the function of Cas9. And so the guide RNA is a long molecule of around 100 nucleotides. There are many possible ways you can change this. How do we figure out how to solve the problem of chemical modification of this construct? Well, working in our lab, we were able to use the crystal structure to identify and make predictions on which parts of the guide RNA we could modify safely and which parts we had to protect. And so through this process, which I will abbreviate in a large way, we came up with a highly chemically modified guide RNA that has improved performance in vivo. And just as one example, we showed that if you create a nanoparticle targeting a gene called PCSK9, you can actually permanently lower cholesterol, hypothetically in patients who have hypercholesterolemia. So in this experiment, we took our chemically modified guide RNAs, a messenger RNA encoding Cas9, we make our nanoparticle, injected a mouse, it travels through the liver, and now the liver is actually specifically edited in this PCSK9 gene. And when we did that, we showed that we could get over 80% of the gene targets knocked out. So essentially all of the hepatocyte genome targets were silenced permanently using this process. And so this was an exciting study where we showed the potential of using these nanoparticles to knock out genes in the liver. And of course, companies have started to pursue this. And very recently, a company called Intellia has evaluated similar technology, which is a modified guide messenger RNA Cas9 LNP in humans. And so what they did is they created these nanoparticles, they injected humans, and actually first this is the primate data they showed earlier back in 2018, where after a single injection, they could get over 60% silencing in primates. And when they looked at, in this case, they're targeting TTR, they could knock it down in the therapeutic range for many, many days. Essentially, it was complete and stable. And so what's really exciting is there's this recent result just disclosed in June where they showed in patients, they could get permanent, well, so far, gene silencing in patients with these lipid nanoparticles. And so when we look at the amount of knockdown that they're seeing of TTR in humans, it's in that same therapeutic range that we saw with the first LNP drug on Patro. However, now the hope is that these patients might just get one injection and that's it. And they're essentially their therapy is provided for the rest of their lives. And so I'm gonna skip the last portion of my talk about circular RNAs, which you're welcome to follow up on if you're interested. And I want to just thank a lot of really smart people that I've had the pleasure to work with. In particular, I want to thank Bob Langer at MIT. I want to thank Yizhou, Gaurav, Dan Siegwart, and also Hyukjin, who were great collaborators of mine back at MIT as well, and my long-term collaborators in companies at Shire and Translate Bio, and more recently at Orna. And with that, if I have any time, I'm happy to take any questions.
Great talk. Now we are going to have some question and answer session. So Professor O will conduct a Q&A session.
All right, so while we are waiting to collect the questions from the floor, okay, now we have some. All right, we have two questions. So first question is, the lipid used for LNP seems to be different depending on the delivery of siRNA, mRNA, and sgRNA. What criteria do you use to synthesize and select the lipids for each type of RNA? Do you have any strategy to optimize the right delivery system?
So the general observation we've had, and I think others have had, is that lipids that are good at delivering, for example, to the liver siRNA tend to also be good at delivering mRNA to the liver. However, if we try to find the very best delivery lipid, often they might not be exactly the same. So in particular, what we find is that the same types of lipids can be used, but the ratios of the different lipids to each other and the RNA can still be optimized to improve performance. So I think that's a long way of saying that lipids for small RNAs and long RNAs tend to be the same, but they also require a little bit of optimization.
All right, thank you so much. And second question is, if the application of siRNA or some mRNA is not for infectious disease or immune disease, maybe it is important not to induce undesirable innate immunity. So do you have any strategy or idea to minimize the undesirable innate immune responses?
Yeah. Well, so it's an important question, and I think there's sort of three answers. First, when you look at the earliest RNA drugs, these therapies were combined with immune suppression therapy before the nanoparticles were given to patients, and that certainly may continue to be valuable depending on what disease you're trying to treat. Second, a variety of work has indicated that chemical modifications can be done to messenger RNA to affect its innate immune response. Now, whether or not the amount of innate immunity that we see with unmodified RNA or modified RNA is significant depends on the dose and the application. And I think one of the outstanding questions as we think about broad application of messenger RNA therapy is in what cases is modified RNA going to be essential, and in what cases might it not be so important? We believe that highly purified messenger RNA that is not modified, that is in fact native, is suitable for many applications in human disease. And so I think we'll have to continue to work on that to figure it out.
All right, maybe the last question from the floor is that it's kind of some opposite the opinion. So how can you modify your delivery system to boost innate immune system? It's kind of the opposite way, but very relevant for mRNA vaccine was so, okay?
Yeah. Well, so one of the things that we've noticed, I'm sure other people have seen as well, is that these nanoparticles can actually stimulate the immune system depending on their chemistry. We, for example, recently published a paper where we developed lipids that could both deliver RNA inside of cells, but also stimulate specific pathways, in this case, the STING pathway, which is a stimulator of interferon gamma. We actually think that the nanoparticles can be engineered to be immunosilent or immunostimulating, depending on your goals.
All right. Okay, thank you so much. I really appreciate your inspiring talk. Thank you.
Thank you.
Okay. So now we are moving to our second speaker, Yizhou. Professor Oh again will introduce our next speaker. Please.
I'm delighted to introduce Dr. Yizhou Dong. Dr. Dong is a professor at the College of Pharmacy and Ohio State University. And Professor Dong, his research interest focus on the delivery platforms of infectious disease, cancer, and genetic disorder. He is recipient of many awards, but if I just introduce only a few, he is a recipient of the Young Innovator Award from the Biomedical Engineering Society and the Maximizing Investigators Research Award from NIGMS, and also he is a recipient of American Association of Pharmaceutical Scientists Emerging Leader Award. So today I'm excited to hear the lipid nanoparticle story of Professor Dong. Professor Dong, please.
Thanks, Professor O, for the kind introduction. Also, thanks Dan for the wonderful talk. He gave a wonderful introduction of the field and also presented many pioneering work for different type of the delivery systems. Also, I would like to thank KAST and Hyukjin for organizing this great international symposium. It's really my great pleasure to share our recent work on the lipid nanoparticles and their applications on messenger RNA therapeutics, gene editing, and also cell therapy. Let me share my screen. Okay. So first, I would like to give you an overview about the research in my lab. We are focusing on developmental platform technologies which can be applied to diverse diseases. The first platform is lipid-like or lipid-derived nanoparticles. We synthesize different type of lipids, and then we can formulate them with different formulation components and also therapeutic cargoes such as siRNA, messenger RNA, or gene editing components. And the second platform, we try to engineer different type of RNA, such as messenger RNA for improved translation or gene editing components for enhancing gene editing efficiency. The third platform is adoptive cell therapy we are recently working on. One example is we can engineer macrophages for treating infectious diseases. And today I'll focus on the first and the third platform. Can I give a wonderful introduction about the messenger RNA-based therapeutics? I'll just briefly talk about this. Messenger RNA has been applied to diverse therapeutic applications in preclinical and also clinical studies, such as cancer immunotherapy, like vaccines, these COVID-19 vaccines, protein replacement therapy, and also genome engineering and gene editing. However, delivery is one of the biggest challenges for the broad application of the messenger RNA-based therapeutics in humans. Dan already mentioned the multiple biological barriers we need to overcome. And to address this delivery issue, we really need to develop different type of the biomaterials. And based on our previous experience on delivery of multiple types of the RNA molecules, we designed this series of TT lipids. It has a long chemical name shown here, and we just give it a short name, TT lipids. These lipids are composed of phenyl ring, 3 amide linkers, and multiple lipid chains. Through this very simple one-step reductive amination reaction, we can synthesize TT lipids in large scale. Also, we can tune the carbon length here from 2 carbons to 8 carbons or even longer chains. This slide shows our overall strategy to optimize the lipid-like nanoparticles for mRNA delivery. And these newly synthesized TT lipids are formulated with helper lipids such as the phospholipids, cholesterol, PAC lipid, and here the therapeutic cargo is messenger RNA. Through this microfluidic-based device, we can assemble these nanoparticles and then characterize the particle properties, such as the particle size, zeta potential, the surface charge, entrapment efficiency of mRNA, and also the mRNA translation efficiency. We utilize the orthogonal experiment design, which is widely used in the field of chemical engineering. This approach enables us to fast identify the optimal formulation with a significantly reduced experiment load. And after that, we can identify the optimal formulation and then test the particles in different disease models. And this is an in vitro screening assay. We formulated TT lipids with luciferase mRNA, and then we treated these nanoparticles with hepatocyte B cells. It's a liver cancer cell line. So on the left, the y-axis, that's the luminescence intensity, x-axis, different TT lipids. You can see TT3 is much better than other TT lipids. Next, we studied the effects of phospholipids on the delivery efficiency. We chose 3 widely used phospholipids, DSPC, DOPE, and POPE. We found that DOPE formulated TT3 is much better than DSPC or POPE formulated nanoparticles. So as I mentioned, we utilize a orthogonal experiment design. We can quickly identify the optimal formulation after several rounds of the optimization process. And also we can analyze the effects of each component on their delivery efficiency. Due to the time limit, I'll just skip that process, and then we apply the optimal TT3 formulation for different disease models. And here is a disease model we chose called hemophilia. Hemophilia is a genetic disorder. It's caused by missing or mutation of factor 8 or factor 9. Patients suffer from life-threatening bleeding or serious complications such as joint bleeds. So in this study, we formulated TT3 with messenger RNA encoding human factor 9. And then we injected these nanoparticles intravenously, first in wild-type mice on the left, the y-axis, that's human factor 9 concentration in plasma, x-axis different treatment groups, including untreated mice, free factor 9 mRNA treated mice, TT3 with firefly luciferase mRNA as a control, and also TT3 loaded with human factor 9 at 2 different doses. You can see it showed a dose-dependent recovery of the factor 9 concentration in the mouse plasma. And similarly, we did a study in factor 9 knockout mice, and this is a hemophilia B mouse model. And similarly, we saw the dose-dependent recovery of the factor 9 concentration. Next, we are wondering if these factor 9 proteins are active or not. So we conducted an activity assay, and here the y-axis, that's human factor 9 activity in MIU per ml, and the x-axis, 3 different treatment groups, control group using firefly luciferase mRNA, and 2 tested group with 2 different doses. You can see the trend is the same. It's also dose-dependent recovery of the factor 9 activity. And at 1.1 milligram per kilogram, the factor 9 activity is around 800 MIU per ml. In normal serum, like the factor 9 activity is between 500 to 1500 MIU per ml. So that means at this dose, we are able to fully recover the factor 9 activity into the normal range in these factor 9 knockout mice. Most recently, we collaborated with a group of Ron Weiss and Daryl Irwin at MIT. We formulated TT3 with self-replicating RNA encoding IL-12. And this is an illustration of the strategy. We did intratumor injection of the nanoparticles. It triggers immunogenic cancer cell death, and then subsequently triggered the anti-tumor immunity. They performed quite a lot of mouse studies and also including mechanism studies. Here are some examples. And in 3 different mouse tumor models, B16F10, CD26, and also tumor 1.7, with a single injection of this LNP formulation, this treatment can significantly extend the mouse survival in all 3 models. And if we delete the CD4 or CD8 T cells, and we found depletion of the CD8 T cells significantly compromised the efficacy of this treatment. Meanwhile, we also did a chemical modification of the DT lipids by installing different type of the lipid chains. And here we have carbon chains, branched ester chains, and also linear ester chains. By tuning these lipid chains, we are able to tune the biodegradability of these lipids. And first, we did in vivo screening of these functionalized DT lipids, and on the left, this is FTT lipids encapsulating firefly luciferase, and we did intravenous injection and measured the luminescence intensity in the liver. We normalized the intensity with TT3, as I mentioned before, this on the left here. And we can see FTT5 is much better than other FTT lipids and also better than TT3. On the right, that's particle properties, the particle size is around 100 nanometer, and the PDI is around 0.1 to 0.3. Next, we studied the biodegradability of these functionalized lipid-like nanoparticles. We chose two different type of lipids, one with branched ester chain and the other one with linear ester chain. So we can see these two lipids with either branched ester chain or linear ester chain, both are cleared very fast from the blood. But the lipids with branched ester chain, they are degraded much slower in the liver, but the lipids with linear ester chain is also degraded very fast in the liver. So by tuning the chemistry of these lipid chains, we are able to fine-tune the biodegradability of these lipid-like nanoparticles. Then we collaborate with a group of Dr. Dennis Sabatino at top. We formulated these FTT lipids with human factor VIII mRNA, and then we injected these nanoparticles in both wild-type mice and hemophilia A mice. We found we are able to recover the factor IX level to a normal range, as we did for the factor IX. And also on the very right, this is the factor VIII activity. We are also able to recover the activity to a normal range. We also did a preliminary study of the histology. We analyzed major organs, including heart, liver, spleen, lung, and the kidney. We found that in this untreated hemophilia A mice, that's control group, and also FTT nanoparticle treated HA mice, there's no significant changes in the histology. Then we collaborate with the company Beam Therapeutics, which is a leading company for developing base editing related therapeutics. So in this study, we formulated FTT5 with mRNA encoding base editor and also guide RNA targeting model gene PCSK9. On the left, here the y-axis, that's a base editing efficiency, x-axis at different doses. You can see it also showed dose-dependent base editing efficiency. On the right, this is one piece of representative data. We injected this FTT5 at a dose of 0.125 milligram per kilogram. We are able to see over 50% base editing efficiency. And our collaborator is very excited with the potency, and to our knowledge, this is the most efficient delivery system for base editing. And currently, these systems have been licensed by multiple biotech companies, which are under development for different clinical trials. Next, I would like to switch the gear to our recent progress on nanoparticle-enabled adoptive macrophage transfer for the treatment of late-stage sepsis. When we design new biomaterials, we try to learn from nature. We always see multiple vitamins in pharmacy stores. Vitamins are natural compounds which are essential for many cellular pathways. Chemically, they possess unique and diverse functional moieties, such as hydrophobic chains, hydroxyl groups, and positive charges. Inspired by vitamin structures, such as vitamin C here in orange, we're thinking maybe we can construct vitamin-derived lipid nanoparticles and apply them to engineer cells for treating sepsis. Sepsis is a life-threatening condition caused by severe infections. It affects over 30 million people worldwide every year, and it remains the #1 cause of death in hospitals. Although the mortality rate reduced with the advance of intensive care and antibiotic therapy, it's still pretty high, about 25 to 30%. This slide shows the challenges for treating sepsis, especially the late-stage sepsis. About 70 to 80% of septic deaths is related to persistent infections. Moreover, multi-drug resistant bacteria have become an additional challenge in the treatment of the sepsis. Meanwhile, these severe infections lead to paralyzed immune systems, such as macrophages. There hasn't been an effective treatment for late-stage sepsis for a long time. Therefore, effective therapeutics are urgently needed. We hypothesized that adoptive transfer of engineered macrophages may be a potential therapy for immunocompromised sepsis. And this is an illustration of our strategy. We designed a series of vitamin-derived lipid nanoparticles to deliver messenger RNA encoding antimicrobial components into macrophages. Then we treated septic mice with the engineered macrophages in order to boost the innate immunity, prevent bacterial immune evasion, and eventually eliminate the multi-drug resistant bacteria. In this study, we first synthesized 5 vitamin-derived lipids, vitamin B3, VC, VD, VE, and VH. And we have these ionizable lipids, which can interact with messenger RNA. And next, we formulated these vitamin-derived lipids with firefly luciferase mRNA, and then we treated with macrophage cell line Rho264.7. And on the left, you can see VC LNP is much better than other vitamin-derived nanoparticles, and also much better than levofectamine 3000 and electroporation. And we also noticed the highest expression of the luciferase is around 12 hours. Then we did several rounds of the formulation optimization, including the formulation ratios and also components. And after that, we identified a lead formulation C5. It's over 7-fold better than the original formulation. It's over 70-fold better than levofectamine 3000, over 300-fold better than electroporation. So we chose this formulation for our further studies. And on the right, here is a cryo-EM structure of the VC-LNP. The particles are in spherical morphology, and it's relatively homogeneous. Then we need to design therapeutic cargoes. Here is antimicrobial components for this VC-LNP. We then studied the mechanisms for the bacterial immune escape. Normally, when macrophage detect a bacteria, they will uptake the bacteria and then form a phagosome. Then this phagosome will be fused with the lysosome to form phagolysosome. Normally, in the phagolysosome, bacteria will be eliminated. But for certain bacteria, they are able to secrete enzymes or toxins and escape this phagolysosome. Then they can reproduce and then infect many other cells. So in this study, we're wondering if we can enhance the antimicrobial activity of the phagolysosome, and then we can eliminate these bacteria in the macrophage lysosome. So in this study, we designed this particular mRNA. It has 3 components. One is antimicrobial peptide, and the second one is the enzyme-sensitive linker, and the third one is cathepsin B. So the rationale is cathepsin B is an endogenous protein of the lysosome. It can bring the whole protein into lysosome. And in lysosome, the enzyme-sensitive linker will be cleaved and then release the antimicrobial peptide, which can help eliminate the bacteria. So in this study, we first would like to check if the cat B is able to bring the whole protein into lysosome. So we produced this reporter mRNA using EGFP. And here is the confocal images. On the left, this is the EGFP signal. In the middle, it's a commercially available lysosome tracker. And on the right, it's an overlay of these two signals. You can see there's a great overlap of the two signals, and the Pearson's correlation coefficient is around 0.91. So that means this protein is able to be translocated into lysosome. And then we did multiple studies using different type of the mouse sepsis models. And I'll just show one example here. And this one, we try to mimic the sepsis patients. So we used mixed multi-drug resistant bacteria as aureus and E. coli. And we first induced immunocompromised condition of the mice. And then we infected the mice with this bacterial. After that, we treated these mice with engineered macrophages. And on the left here is quantification of the bacterial load from the mouse serum. And the one group is PBS, the other group is unengineered macrophages, and the third group is engineered macrophages. You can see engineered macrophages significantly reduced the bacterial load. And more importantly, these engineered macrophages dramatically extended mouse survival. And here, over 80% of the mice survived, and they are fully recovered. And then we characterize the biomarkers. One is body weight. We can see all the survived mice continue to grow. And also after 30 days, their white blood cells and lymphocytes both return back to the normal range. So in conclusion, we first talk about lipid-like nanoparticles. We showed a proof of concept study to deliver multiple types of mRNA, and we also tested these different types of nanoparticles in disease models, including hemophilia, cancer immunotherapy, and also for base editing. And for the second part, we talk about adoptive macrophage therapy. We identified vitamin C derived lipid nanoparticles are particularly great for mRNA delivery in macrophages. And then we engineered these macrophages. They are able to improve the survival rate for the septic mice, and they clear the bacteria from the mouse serum, and eventually they restore the body condition of these septic mice. So in the next few years, we really would like to utilize our specialty together with our collaborators in the fields of the biomaterials, RNA engineering, genome editing, immunoengineering, and cell therapy. Our ultimate goal is really to translate our technology development and research findings into more effective treatments for the patients in the clinic. Lastly, I would like to acknowledge many people. First, we have a group of talented postdocs, graduate students, undergraduate students, and several high school students. And also thanks our collaborators for their significant contributions to many projects. And thanks funding agency supporting us. Thank you very much for your attention. I would like to take any questions. Thank you.
Thank you, Dr. Dong, for a very stimulating talk. So we have some questions from the floor. So first question is, did you modify mRNA chemically to improve the stability? If not, what is your strategy to improve the stability of mRNA?
Yeah, that's a great question. I didn't mention about that. We have one direction, particularly engineering messenger RNA. One approach is to utilize chemically modified nucleotides. And for our messenger RNA, we do use chemically modified nucleotides. And another strategy is to engineer the UTR, untranslated region of the messenger RNA. Both can significantly improve the property of the messenger RNA.
All right. Can you reveal what kind of chemical modification if it's not patented? Well, it's a secret.
Yeah, we have done a particular study, explored multiple chemical modifications, and in that study we identified the chemical modifications. One is a pseudo-U, the other one is a N1 methyl pseudo-U, and the last one is 5-MOU. So these are three we normally use for chemical modifications.
All right, thank you. So the second question is, when you deliver mRNA machines with delivery system, how did you test the stability of mRNA in vivo system? How did you measure the integrity of mRNA?
So for that perspective, we can do RT-qPCR to analyze the half-life of the mRNA.
All right. Okay. So quantitative RT-PCR, maybe, right? Yeah.
Yes.
All right.
Yes.
Okay. All right. Okay. So, in your sepsis study is very interesting. And one question is, what is your strategy to directly target macrophages? Are there any targeting ligands to macrophages?
No, we didn't use any targeting ligand. We just found that the vitamin C lipid nanoparticles are really great for delivering mRNA into macrophages.
All right. Okay. Now, your vitamin stories, you actually screened several vitamin derivatives. And what do you think is the mechanism by which vitamin C derivative significantly showed the highest delivery efficiency? Is there any theoretical background?
Yeah, we did some preliminary studies. We found, if I remember correctly, the caveoli-mediated endocytosis is a major pathway for cellular uptake of these vitamin C-derived lipid nanoparticles.
All right. You actually modified mRNA chemically, and I think maybe there are some reports that that kind of chemical modification can activate innate immune system sometimes. So for your application for sepsis treatment, is there any risk related to exacerbating the sepsis symptom by the chemical modification of mRNA?
That's an interesting question. In fact, I think for certain chemical modifications, they can significantly reduce the immunogenicity of mRNA. So in our study, we used chemically modified nucleotides. We try to minimize any immunogenicity from either the messenger RNA or the nanoparticles. We hope we can just deliver the therapeutic cargo, that is the antimicrobial components, into messenger RNA, just to enhance the antimicrobial activity in the macrophage lysosome.
All right, and maybe the last question. Your talk is very much interesting. So maybe the last question is that have you or any other research group that you know have developed or used computer simulation program to design functional new lipids or polymers for your LNP lipid nanoparticle, like some kind of AI-based technology or computer simulation? Do you know any other groups?
Yeah. I think there's already published papers. I think also there's companies. I think both academic and industry are interested in that. And we recently published a review article. We think it will be a very important area to explore.
All right. Can you tell us what kind of which journal and promoting your review article in here?
Sure. Thank you. Yeah. Quick look. Yeah, it's a collaborative review article. 1 second. Let me try to.
Okay, we will search.
It's a collaboration with computational scientist Dr. Xiaoling Cheng, and this article is published on current topics in medicinal chemistry.
All right.
The title is Computational and Experimental Approaches to Investigate Lipid Nanoparticles as Drug and Gene Delivery Systems.
All right, thank you so much. It is really interesting talk, and many people are enthusiastic about your talk. Thank you very much, Dr. Dong. All right, I will
My
conclude.
pleasure.
Okay, thanks.
Thank you. Thank you.
All right, now I'll move on to the third speaker of this session, Dr. Daniel Siegwart. So Dr. Siegwart is an associate professor at the University of Texas Southwestern Medical Center. So Professor Siegwart's lab, his research interest includes the delivery of short and long RNA for gene silencing, gene therapy, and gene editing. And today, Dr. Siegwart will share with us very interesting topic about ionizable phospholipid, one of the key elements in that kind of nucleic acid delivery. So please welcome Professor Siegwart. Dan?
Thank you very much. Great. Thank you very much to the Korean Academy of Science and Technology. It is an honor to be invited, and I greatly appreciate the opportunity to share our research with this audience. It may be hard to think about today, but in not too distant future, we will look back at the end of the COVID-19 pandemic and think about how vaccines are the thing that brings us out of the current situation that we all find ourselves in. I believe that we also look back and consider 2020 as one of science's greatest years in human history. Typical vaccines take decades to discover and develop and deliver around the world. And here we are with multiple vaccines that are effective against the SARS-CoV-2 virus being developed in really less than 1 year. This is just a remarkable scientific achievement and is the way out of the pandemic to where we will be able to someday, and again, not too distant future, be able to socialize again in person and communicate and share ideas, just as KAST is having us do today virtually in person. As we know, two of the leading vaccines are messenger RNA lipid nanoparticle vaccines designed to encapsulate messenger RNA encoding for the spike protein of the SARS-CoV-2 virus in the lipid nanoparticles that are then injected into the muscle. These nanoparticles transfect muscle cells and antigen-presenting cells that are recruited to the site of the injection to train the immune system to fight against potential future infections of the SARS-2 virus. These vaccines are incredibly effective. And when we think about what work has been done in this past year, it really is remarkable. The sequence of the SARS-CoV-2 virus was published publicly on January 10, and just 63 days later, Moderna, in collaboration with the National Institutes of Health in the United States, started the first human clinical trial for the vaccine that many of us have taken or will be taking in this coming year. So it's really amazing, and it feels like it came out of nowhere, and it also feels sometimes like, why didn't we do this before? So I liked reading this article in The Onion. The Onion is a satirical humor magazine in the United States, where they published an article of the title, "Nation Can't Believe They Spent So Long Overlooking Obvious Solution of mRNA Instructions for Spike Protein Encapsulated in Lipid Nanoparticle. It's so obvious in retrospect, say 327 million pandemic-ravaged Americans." And indeed, it may feel this way. And I understand that many people in the public have concerns about how fast this vaccine was developed and how safe might it be. Therefore, I would like to provide some comfort in that this is a technology and an approach that is resting on decades of scientific research. Messenger RNA was discovered in 1961. Liposomes, which are the precursors to lipid nanoparticles, were developed in 1960s and 1970s. Katie Carrico and Jonathan Weissman showed in 2005 that nucleoside modified messenger RNA is non-immunogenic. Dan Anderson, who you've heard from already, Bob Langer, Peter Collis, and others developed the modern concept of lipid nanoparticles, really going back 20 years. CRISPR has been discovered and shown to be effective in humans. So really the development of these vaccines is resting on all of this knowledge. And it gives me, and I think many others, great comfort in receiving these vaccines, that these chemical structures and this technology has been used in humans before and is a safe and effective approach. I quickly downloaded the labels for all of these vaccines and was almost delighted to find that the recipe for the Moderna and Pfizer vaccine looks very similar to the recipe that Alnylam used in Onpattro. This is the US FDA approved drug called Onpattro for silencing TTR that Professor Anderson mentioned, composed of lipids that are not that dissimilar from the lipids used in LNPs. So today I would like to start by just introducing this concept and appreciate the invitation from the Korean Academy of Science and Technology to think about what's being done today and what will be done in the future, to look forward to the future genetic medicines that may be a part of our lives in disease treatment and in vaccines. One of these futures is in genome editing and genome correction. Just a few weeks ago, Intellia reported using liver targeting formulations that are not that dissimilar than the ones on the previous slide, that CRISPR-Cas-based gene knockouts are functional in both non-human primates and in humans. These are also highly durable medicines that so far have been safe in the sense that they are not editing off-target sites in humans. This indeed is one of the future ways and provides a compelling case for the treatment of genetic diseases. So let's think now for the rest of the presentation about what may be coming in the future. We can understand that current generation nanoparticles are effective vaccines. They are effective at delivering nucleic acid contents to the liver, examples being Onpattro and the aforementioned vaccines. But as we know, disease-causing mutations for cancers, for genetic diseases, for a variety of different ailments that plague mankind occur in all different tissues and all different cells, from the CNS to the heart, to the kidneys, to the lungs, to the spleen. Therefore, I feel it's incumbent on material scientists like us to discover and develop ways to deliver nanomedicines outside of the liver and inside of targeted cells. So today I'll talk about two strategies that allow one to, using rational design, redirect nanoparticles to extrahepatic tissues, and using rational design, increase the ability of those nanoparticles to get out of endosomes and release the therapeutic drugs inside of targeted cells. So first, to set up this concept, I want to provide a couple of observations that might even answer a few of the questions asked so far. So the first is around, could we use the same molecules, these cationic or ionizable aminolipids, phospholipid, cholesterol, and pegylated lipids that go into a recipe, a nanoparticle formulation for siRNA, for a messenger RNA? And we and the other speakers in this symposium have found that the answer is generally yes, but it can be optimized further by adjusting the proportion, the molecular amounts of these molecules within each LNP to increase potency, to control the size and architecture of the nanoparticle in a well-controlled way that promotes endosomal escape. So the first observation is that existing lipids can be used to deliver long and short RNAs, but that the way they package around RNAs inside the LNP is a critical component to their efficacy. The second observation I want to share was inspired by a computational model looking at those relationships between RNAs inside of nanoparticles. Here we hypothesized that combining the zwitterionic phospholipid with the ionizable cationic lipid may improve local interactions of solubilized RNA, potentially initially in water pockets within LNPs, to increase activity and delivery of longer or structured folded nucleic acids, like transfer RNAs and messenger RNAs. Indeed, we found that to be true. And moreover, we noticed that upon delivery of luciferase encoding messenger RNA or genome editing of a reporter mouse, that these formulations could achieve editing in the spleen, lungs, liver, and kidney. Although we could not control the specificity of delivery at that time, it gave us some clues as to how to devise that into a programmable and predictable strategy for tissue-specific messenger RNA delivery. And the final observation to share rests on this premise that one may be able to use chemistry and chemistry alone to control which cells take in a nanoparticle. And one example of that, we used a high throughput library of functional polyesters that had a very high degree of chemical diversity and screened them for the ability to deliver siRNA to normal and cancer cells derived from a single patient at the UT Southwestern Hospital just outside of my window. So this patient donated her cells. The normal cells were immortalized so that we could culture them side by side. This study led to the identification of a nanoparticle that purely uses chemistry to internalize in cancer cells, but not normal cells, and stay in the tumor for weeks, as opposed to being diffused out in a couple of hours. In other words, it showed that chemistry alone can dictate an endogenous identity that promotes selective uptake into cells. So here in this first example, I want to highlight the work of Chong Chang and Tuo Wei. They will both be starting their labs in Beijing very soon. And we've asked this question, could we use chemistry to control tissue tropism? This is a platform we call selective organ targeting, or SORT. It allows for redirection of LNPs to extrahepatic tissues in a predictable and programmable way. It starts with the traditional LNP that we've heard a lot about in this symposium. These molecules, as well as the amounts of these molecules, are essential to form efficacious nanoparticles that internalize in cells, release their nucleic acids to be translated into functional proteins. We reasoned that if we made other molecules that looked exactly like these molecules, that this approach would improve liver delivery, but may not allow us to discover formulations that go outside of the liver. Therefore, we hypothesized we could add molecules that normally do not belong in LNPs. They may have chemical structures that are outside the box of a conventional 4-component LNP design. In doing so, we discovered that classes of molecules that harbor quaternary ammonium groups allow for now 5-component LNPs to be redirected to the lungs, in this case, delivering luciferase encoding messenger RNA. They deliver the RNA from the blood side all the way to the airway side. In green are anionic lipids and molecules that are not necessarily lipids, but harbor specific chemical functional groups that redirect LNPs to the spleen, where they can deliver to cells like T cells and B cells. And we could add other molecules that promote a cell expansion in the liver so that they could deliver to twice as many cells and produce more than 10 times the amount of therapeutic protein. So let me explain a little bit more how this was discovered and I think what it means for delivery of genetic medicines. Chong was very great. He was willing to do this study on the first day. We decided that if we would make a random formulation, we may not be able to understand the structure activity trend. Therefore, we made formulations that had 5, 10, 15, 20, up to 100% of additional supplemental molecule. We called a SORT molecule. And then we imaged these mice after delivery of this luciferase encoding messenger RNA intravenously after 6 hours. And we saw that the delivery started in the liver, moved to the spleen, and at high percent incorporations now went almost exclusively to the lungs. This gave a way to see that it's programmable, it's dose responsive, and at high incorporation, like 40 to 50%, we now have a lung-specific formulation. In that example, we picked DOTAP because it had been widely studied in the literature and lipoplexes. But we've now looked at tens or hundreds of these molecules and found that it is generalizable to a class of structures. Here are 2 of them, DDAB, that's quite simple, and EPC, that's a bit more complicated. But it shows the same progression pattern, allowing for lung delivery at 40 to 50% incorporation. We next asked what would happen if we introduced the opposite characteristic into the LNP. And here is one example, a lipid termed 18PA, that when introduced from 5 to 40% into the LNP, now redirects expression, the mRNA translation to protein, exclusively in the spleen. Interestingly, we found that we could supplement our 5A2SC8 4-component LNPs with other ionizable aminolipids like DODAP, or even more 5A2, and typically at 20% incorporation that increased liver delivery. Therefore, SORT is this way that we could take established formulations and redirect them to different tissues. To test that, we downloaded the label for Alnylam's Onpattro, which is this normally siRNA lipid nanoparticle. Instead of siRNA, we encapsulated messenger RNA and saw the expected liver targeting and liver expression of that protein. Now, by introducing SORT molecules into this established formulation, one can redirect expression to the lungs or to the spleen. And we're now working on additional tissues that will be forthcoming in future publications. SORT can be used for genome editing in addition to messenger RNA like protein replacement therapies. One way to test that is to use this activatable mouse model, wherein the mouse would be expressing a red fluorescent protein, but it's held back by a lock-stop-locks TD tomato cassette. One could introduce Cre recombinase messenger RNA or a CRISPR or base editing designed to cut the locks and delete the stop sign and then turn on red fluorescence. It gives us a way to detect tissue-specific liver, lungs, or spleen delivery and quantify the activity or the editing efficiency in different cellular populations. For example, using confocal microscopy, we can detect red positive cells in each of these organs. And by flow cytometry, we can quantify what percentage of, say, all hepatocytes are gene edited and find that liver sort edits over 90% of cells. What percentage of all epithelial cells or endothelial cells are edited. And interestingly, lung sort can edit both the endothelial cells that line the blood vessels and the epithelial cells that line the airways. We're now finding that therapeutically attractive cells in between are edited too, including basal progenitor stem cells, and are working towards correction of this lung diseases using these lung sort formulations. And finally, with spleen sort, there are attractive cellular populations, including T cells and B cells, that may be compelling for endogenous CAR T cell generation or B cell lymphomas. And finally, for this section, I'll highlight the efficacy in targeting the liver as one example. We're obviously working on lungs and spleen and gene correction in multiple tissues. But here is this target PCSK9 that's been mentioned already. And liver sort formulations can target PCSK9, induce indels at about 55 to 60% frequency. And that leads to complete loss of PCSK9 in both the liver, where using a Western blot of homogenized liver tissues, we cannot detect PCSK9. And by ELISA, we cannot detect any PCSK9 in the serum of these animals. I think that this, as well as the other presentations you've heard today, indicates that this is a viable strategy to take forward into the clinic. So we've talked so far about how do we design lipid nanoparticles. How do we design nanoparticles that may go to the liver if that's the target, or avoid the liver and go to other parts of the animal, like the lungs or the spleen, the bone marrow, the lymph nodes. So in this final section, I would like to comment on once the nanoparticle reaches the cell, are there ways to use chemistry to improve its ability to unpackage its contents, the therapeutic drugs, and release them into the targeted cells? And this was a project led and finished by Shuai Liu, who will also be starting a faculty position soon. There are many barriers to intracellular delivery, and these barriers are quite significant when thinking about next generation technologies like CRISPR-Cas-based gene editing. CRISPR for genome correction via the homology-directed repair mechanism would require a Cas9 messenger RNA or protein, a single guide RNA, and a donor DNA template that may be encapsulated in a single or multiple formulations. Each of these contents need to be released into cells, likely by endosomal escape. So we've recently systematically studied that, and if you're interested, there's a paper that came online in Advanced Materials. But I'd like to focus in on one of these potential mechanisms. This is the plausible mechanism of membrane destabilization, that the chemistry of the molecules that are in LNPs may fuse with endosome membranes to allow for intracellular release of encapsulated RNAs. So for this project, we were inspired by historical literature around polymorphic phase transitions that lipids can undergo. So lipids are really interesting molecules with polar groups like a polar head and hydrophobic domains like alkyl or alkenyl tails. And depending on the characteristics, such as the size and shape of the head or the body, depending on those properties of the lipid, lipids can undergo micellar to bilayer to hexagonal phase transitions. And these phase transitions may be exploitable to allow to increase endosomal escape. So the rules for many of these parameters have been worked out in the fundamental literature, and it's been such a pleasure to read that, wherein shape factors and other fundamental parameters can be used to identify, based on the chemistry, what these molecules can do. As one prototypical example, we can think about DLN-DMA as a lipid with a small head group, simple linker, and 2 hydrophobic domains. It has been shown that this lipid is neutral outside of the cell, because the head group is not charged. It has a pKa about 6.4. But inside of endosomes, where the pH is lower, this nitrogen becomes positively charged. So it's neutral outside of the cell, positive inside of the cell in endosomes at the maturing endosome stage. And that charge acquisition might support the shifting of the structure into the cone shape, because that positively charged lipid can associate with negative lipids in the endosome and induce this shape change that might allow for increased fusion with endosome membranes and release of the RNA contents into cells, a mechanism that has been proposed some time ago. So we decided to see if we could put a twist on this and make molecules that are honestly a little goofy in their conception. Most lipids for RNA delivery are neutral outside of the cell and positive inside of the cell. We wondered if we could make a lipid that would be negative outside of the cell with an ionizable amine and a negative phosphate group. And then inside of endosomes, the nitrogen would be protonated to induce the formation of a net neutral zwitteran. So here they would become neutral inside of the endosomes. So why would we do that, and how would that work? Well, we hypothesized that if we made a molecule that had a small head and a big body, that induction of this phase transition would create a molecule like this that, due to its zwitteranic nature, could mix with lipids that are in membranes, like endosomal membranes, and then induce a lamellar to hexagonal phase transition. So it would be a twist on the chemistry of endosomal escape by making a molecule that essentially becomes a zwitteran inside of cells. And that small head and big body might be critical to create a structure like this and make the cone shape. Okay, so to access that, we needed to develop some new chemical reactions. And to do that, we made a series of alkylated dioxyphosphorylane oxide molecules, essentially a structure that could undergo ring opening reactions to make one part of the zwitteran. And here, Schweiss systematically made all different hydrophobic domains to test the effect of the alkyl length of this part of the molecule. With these in hand, we could do ring opening reactions with a variety of different amines and make complex architectures that might have one or multiple branches, one or multiple switchable amines, one or multiple zwitteranic heads, big body, small head, big body, so on and so forth, and test their activity and look for structure-activity relationships. We examined delivery of luciferase encoding messenger RNA to an IGRAV1 ovarian cancer cell line, and found that the activity largely concentrated in 2 domains, one being here, horizontally, and the other being here vertically. This was good news in a way because it allowed us to identify very clear structure-activity trends. The first trend was that molecules with a single zwitterin were much more likely to be active, higher hit rate, than molecules with multiple zwitterins. This was supportive of the idea that a small head would be important for that phase transition. We also found that 3 hydrophobic tails was the optimal structure. There were some hits with 4 and 0 hits with 2, implying that multiple tails and a big body was important for that phase transition. And finally, the tail length, as is true for most all lipids, did affect activity with an optimal tail length between 8 and 12 carbons. Next, we wondered from prototypical molecules, could they actually undergo that phase transition? So we used phosphorus NMR to study whether this IPHOS molecule, we called ionizable phospholipid, could mix with model endosome membranes and induce the bilayer to hexagonal phase transition. And indeed, we saw the characteristic peak shift that is representative of that fundamental biophysical property. We also performed a series of studies, including hemolysis of red blood cells, assays at different concentrations of different molecules, and at different pHs to test whether these molecules could induce membrane disruption at low pH and not high pH, which was indeed the case. And finally, we used a variety of different FRET-based assays to test the ability of iphospholipids and iphospholipid nanoparticles to destabilize membranes, including model endosome, lysosome, and cell membranes, to demonstrate their ability to specifically destabilize endosome membranes, which they did. We also performed in vivo analysis of RNA delivery. And this was really interesting because we found that there were a large number of structures that could be formulated into self-assembled lipid nanoparticles to achieve RNA delivery in vivo after intravenous administration. And there were also interesting structure activity trends. For example, the functionality at the amine side was the largest driver of the efficacy, that specific amines were more active than others at allowing for that endosomal escape and in vivo delivery property. So the amine side of this 3-part molecule was largely controlling the efficacy. On the other hand, on the phosphate side, the hydrophobic domain at this phosphate side controlled the tissue specificity, where shorter alkyl domains led to liver delivery and longer alkyl domains led more to spleen delivery. Therefore, we're having fun thinking about this as a 1, 2, 3 component molecule, wherein each part of it could control efficacy or tissue tropism based on the chemistry. And maybe in the final slide or two, I'll just illustrate how these molecules could be used. One way to think about them is that they are like a helper lipid. So they could replace DSPC in a 4-component LNP or replace DOPE. And indeed, they are more active at delivering RNA in vivo. So they could be thought of as a helper lipid that boosts endosomal escape in vivo. Alternatively, iphospholipids could be combined with other phospholipids. In that case, we found this drove spleen delivery of RNA. iPhos could be combined with ionizable aminolipids that generally promote liver delivery. And indeed, in that way, we can make more efficacious liver targeting formulations. And finally, we can mix iphospholipids with permanently cationic lipids to drive lung delivery. And in the paper and in ongoing work, we've been able to do things like genome editing by delivery of Cas9 mRNA and single guide RNA to delete this stop and turn on the red fluorescence to correct genetic disease mutations in multiple organs. And we're excited to put this into the toolbox of formulations that we use in the future. So with that, I'd just like to conclude by explaining this way of using chemistry and engineering to understand what's happening inside of the LNP and outside of the LNP. And I've shown today 2 approaches, one to control where the nanoparticle goes and which cell type can be transfected, and the second to focus on what happens inside of the cell to use lipids that are rationally designed to increase endosomal escape and make next generation more effective, more safe delivery vehicles. Building on the success of RNAi and now these LNP COVID-19 vaccines, I'm sure that there's a bright future ahead for a variety of genetic drugs in our lifetimes. Would like to acknowledge funding from various agencies, the Korean KAST for a wonderful invitation and a wonderful symposium, and the lab members, including Shuai Liu, Chong Chang, Tuo Wei, and many others who did this work. This has been one of the bright spots of the last 1.5 years for me, has been the increased interactions with my lab and seeing them persevere through the challenges that we've all experienced to just do more work and publish more papers and contribute more to humanity. Thank you all for your attention, and I'd be delighted to take questions.
All right, Dr. Siegwart, I really enjoyed your talk. There are several questions from the floor. The first question is, for Intellia study, how come the human clinical data with 0.3 milligram per kilogram dose give the better result as compared to non-human primate data, which is administered at the dose of 1.5 milligram per kilogram? Do you have any explanation for this dose-dependent efficacy issue?
Yeah, I think it's a good question for the company to answer directly. Some things that come to mind for me from an outside point of view. One is that they do design the guide RNA to have very specific chemical modifications that increase the activity and the specificity. So in the human study, there could be advantages of that specific single guide RNA. Additionally, there are different concentrations of serum proteins in non-human primates than there are in humans. And the one that is likely involved for liver-targeted formulations, including Intellia's LNP, is apolipoprotein E. So that may be a factor that the PK/PD of LNPs are a little different in non-human primates than humans. The guide RNA may be a little bit better guide RNA. And there may be some endogenous targeting effects that give an extra boost in the humans. It's overall great news, I would say, particularly for the future of genetic diseases of the liver.
All right, thank you. And your whole talk is about ionizable phospholipid, but for the beginners in this field, can you underline what is the unique advantage of ionizable phospholipid as compared to conventional cationyl lipid-based cationyl liposome?
Yeah, I think throughout the history of nanomaterials for RNA delivery, the nucleic acid is negatively charged because it has the phosphate backbone, whether that's double-stranded siRNA or microRNA, or single-stranded messenger RNA or DNA. These molecules have negative phosphates. Therefore, one uses cationic, positively charged amines to bind those negative RNAs and allow them to self-assemble and encapsulate inside the nanoparticle. The pH switch, whereby the amine can be neutral outside of the cell and positive inside of the cell, is also a critical hallmark for delivery of nucleic acids. In terms of inspiration, I think there should be ways to use molecules other than amines to do that step. And with HiPhos, it was one of these moments of just what would happen if we made this kind of science? What would happen if we made a molecule that did not use charge acquisition to become positive, but rather use charge acquisition to become neutral and physically change the shape? So the shape changing is the unique aspect. Is that better than using pH to just change the neutral to positive? I'm not sure. I would say today that this is a new tool to put into the toolbox and combine with other molecules and see what would be possible with further chemical development.
I see. So you think that the shape change may help to promote some endosomal escape and increase the efficiency? Is it all right. All right. And there are some questions about the organ-specific delivery. That is very interesting story for your SORT story. So in your opinion, do you have any explanation or some underlying mechanism for explaining for the specific organ targeting? For example, why do you think the 18PA LNP shows spleen tropism? Something like that. So some mechanistic your insight for the organ targeting.
It's a great question. We have submitted a manuscript on this topic, so hopefully it'll be publicly known soon. Layers of the mechanism for extrahepatic targeting. I think what I can share now is that there are some guidelines for liver delivery, and it appears that lung sort and spleen sort and other sorts do not obey those commonly assumed guidelines for RNA delivery. So I apologize for I guess protecting the work of the investigators who stay in the lab all day and all night, like it is here in Dallas. But stay tuned, there'll be some information forthcoming about the detailed mechanism.
Okay, I look forward to your forthcoming the paper. And my last question that I have is that you observed some kind of the molecular ratio dependent lung targeting, but does lung targeting mean that it does not mean that it is actually delivered to intracellular of some specific cell? Do you have any idea in terms of lung targeting, what type of lung cells are really prone to actually take up that kind of your RNP, cationic RNP, ionizable one? Yeah.
Sure. We have been able to demonstrate and quantify cellular uptake and RNA translation to the protein and genome editing in multiple cell types of the lungs, including endothelial cells, epithelial cells, basal cells, and macrophages. So that is a pathway to go from the blood side throughout the lungs.
I see. So is it kind of what kind of interaction? Is it some kind of receptor-mediated one?
We believe there's a receptor-mediated interaction component to the process, yes.
So some unknown receptor so far, which promote the delivery into the cell so far, all right? Okay, and one more question just from the floor. So so far you showed the kind of organ targeting to liver, spleen, and lung. But what do you think about the some specific delivery to other organs, such as brain, for CNS disease, genetic disease treatment? Yeah, that kind of application. Have you thought about it? Yeah.
For brain, there is the blood-brain barrier that would have to be overcome. Therefore, I feel this will remain a challenging area for nanoparticles. Professor Anderson talked well about the organs and tissues that are accessible from the blood. So the BBB is a one blocking component. I would say it's not possible and not impossible, and also may not be necessary. For example, Novartis has an AAV drug that is intrathecally administered, and that's an FDA-approved viral vector that can be used to treat a genetic disease of the brain. In terms of what's next, I think that bone marrow, lymph nodes, and hopefully some of the other systemically accessible organs like the kidney, maybe the pancreas, maybe the next organs that could be achieved. And I hope that lots of people work on this, and there are a lot of great solutions to this problem because there are plenty of cancers and diseases in great need.
All right, thank you so much. Now I will let you go. Thank you for the enthusiastic talk. Thanks.
Thank you.
All right, now I'll move on. The last but not the least speaker of this session, Dr. Gaurav Sahay. Dr. Sahay is an associate professor of College of Pharmacy at Oregon State University. His research interests include the delivery of messenger RNA therapeutics for treatment of lung disease such as cystic fibrosis and infectious disease, very hot SARS-CoV treatment. And he is the recipient of many big society. For example, he is the recipient of the Controlled Religious Society Nagai Award, and he is the recipient of the American Association of College of Pharmacy New Investigator Award. Today, Professor Sahay will share with us the very interesting topic about intracellular delivery of mRNA. Please welcome Professor Sahay.
Thank you so much for the very kind introduction. I think it's my pleasure to be in this KAST symposium, to all the organizer and Hyukjin for this invitation. I've listened to some really great talks, so this is a really difficult proposition. But what I did was I managed to get a blue shirt because I see like all my friends had a blue shirt, so I thought I'll be in the club as well. So the topic for me today is intracellular delivery of messenger RNA, and I hope you could see these slides. So the field of mRNA therapeutics, I think, has moved from a renaissance to an industrial revolution. What began in March with the initial clinical trials to things that are actually very surreal because they're happening on the front pages of New York Times, where how to dose patients, how much would be the dose, to things like stability, which is one of the key components of pharmaceutical sciences, is being discussed. So the impact of nanotechnology and drug delivery, I think for students out there, is clear, especially during these times where a lot of us have been now injected with some of these technologies and how fast they have moved. So from the perspective of how fascinating this has been, for the new trainees, I think that's a very exciting time to be in. Before COVID, we used to talk about mRNA delivery, where there was no risk of genomic integration, does not need to go to the nucleus, rapid and transient effect. So one thing for vaccines definitely is you need a transient effect, the antigen gets produced and goes away. So it was a perfect application. Before COVID as well, a lot of people were working on therapeutics, but the issue there, which still I think we need to resolve, is how to do a repeatable dose that maintains the level of protein expression. But vaccines was something which could be moved very quickly, and we have a real world sort of view of that at this point. The challenges are, as a lot of my esteemed friends and colleagues and mentors have talked about, is the challenge of delivery of mRNA. It's a negatively charged molecule. It is unable to enter the cells by itself, and it's highly susceptible to degradation. Having said that, as again has been talked a lot, and I'll just briefly touch upon it, that the enabling technology, one of the heroes of this story is also the lipid-based nanoparticle, which can encapsulate the RNA, and then once it's encapsulated, it has to go in and deliver. So you can make a lot of particles which can encapsulate the material, the cargo really well, but there are only very few which end up going to the cell type of your requirement. And Dan and Yizhou and Dan Anderson all gave a fantastic talk of how much it's required in terms of engineering to enable these technologies to actually end up in the cell and do its job. And one of the key components here, all of these topics have also been touched, but I'll also say that one advantage of lipid nanoparticle is that you could actually give large cargoes. AAVs have short cargo capacity, things like CFTR, you cannot deliver in an AAV. You can have just one shot of an AAV rather than multiple doses. And then scalability has now another sort of definition, because the companies like Moderna and Pfizer are scaling these technologies for billions of people. So if you find a lipid nanoparticle that can deliver to the brain, for say, or the bone marrow, it would be very easy to scale it, because the next steps, which was usually a challenge in the drug development process, is now being cleared. I think there'll be newer way of manufacturing these materials. Right now, we are in like generation 1. 5 years from now, we'll have different ways of manufacturing to the scale. But I think it is a phenomenal time, because once you identify these enabling technologies for rare disorders, you could supply these for the rest for the rest of the life of people. And modularity, again, which has been talked before, from sRNA to gene editing, there's a huge ability of these materials to deliver a lot of different cargo. So the first LNP that was approved was on Patro, and then came the mRNA vaccines. And if you could look at it, they are composed of the ionizable lipid, and then DSPC. The differences are very minor. I mean, if you look at the ionizable lipids for Pfizer or Moderna, and then the other difference is the PEG lipid is a little different. But whole and soul, once you inject these mRNA vaccines in the immune system, they possibly go to the lymph nodes or the dendritic cells, and that leads to activations of a lot of different immune cells in the body. And we have a review that appeared in 2021 where we talk about a little bit about these vaccines. And the results have been dramatic with both these products. You see 95% efficacy. Both have, I talked a little bit about the LNPs, but the mRNA is also a little modified because it's the spike protein, but it has 2 proline modifications so that it remains in its pre-fusion configuration. And that has seemed to be a really potent way of getting this vaccine to work. So as Professor Anderson was saying, sequence and the sequence target, that 2 proline modification was important. And then the different lipid components that can actually hit the immune population was really critical and has helped in developing some of these vaccines. So where do we go next? I think, and Dan Siegwart had talked a little bit about it, that we, I think possibly now would be moving into different organ systems like the lung, the pancreas. I'll talk a little bit about our work in the eye and delivery to the eye. Could be possible to deliver to the brain. And I think extrahepatic delivery of these lipid nanoparticles for the next 5 to 10 years will transform the field of medicine, from genome delivery of genome editing components to gene delivery. And I think that's a very exciting time to be in. So I'll talk a little bit about how these particles get internalized inside the cell. So we listen to great talks of how these particles are formed, or what happens once they interact with the cells and internalize inside the cells. So after an IV administration of a typical LNP, ApoE binds the surface of the LNP, the PEG lipid desorbs, and then ApoE starts to bind. And the structure of these LNPs is still something which people are trying to figure out. These are multi-component lipids. Where is which lipid present in the LNP is something that is also being researched in academia as well as industry. So ApoE binds to the surface, takes it inside using the LDL receptor. From here, things start to, I think, disintegrate a little bit in the lipid nanoparticle, where then the ionizable lipid interacts with an endosomal membrane. And the escape is for sRNA is less than 2% in vivo. Moderna had a paper where their lipids showed less than 15% efficacy. They're efficacious in terms of delivery because a lot of these things are enzymatically driven, but there is still a lot of material waste happening. So engineering novel materials that can cross these endosomal barriers remains important. And when I was a postdoc with Professor Anderson, we also figured out that there is a cholesterol transporter called Niemann-Pick type C1, which recycles these particles in vitro. So based on that there is a cholesterol transporter that recycles some of these materials, we started to collaborate with Moderna in around 2017, where we started to look at how different cholesterol analogs can be used in place of the regular cholesterol. So you heard about a lot about helper lipids, ionizable lipid, but what happens when you change the cholesterol? And what we did was we grouped these cholesterol analogs, and these are naturally occurring, so you can buy them off the shelf based on differences in the body of the cholesterol structure, the tail or the head group. And based on that, we formed 3 groups. what we found was that C24 alkyl derivatives of cholesterol, beta cytosterol, had enabled similar encapsulation and size, but the transfection efficiency in vitro could lead from, like, based on the cell type, from 30 to 200 fold. So only by changing the cholesterol analog, we were seeing a boost in gene delivery independent of the ionizable lipid that we were using. Either it was like lipid 9 from Moderna, or we were using some of the MC3 based materials. And what we then looked into is what was happening here. What did cytosterol, the C24 alkyl derivative, and once we plugged cytosterol in, we call it as an ELNP. So it has all the same components, just replacement of cholesterol. Cytosterol had the particles that contained cytosterol, or ELNPs, were a little larger in size, but what we saw was these polyhedral shapes that arised in these ELNPs. And the internal structure, using the SAX analysis, we found little or no difference there, but it was the exterior that got changed and these polyhedral shapes were formed. When we looked at other C24 alkyl derivatives, they all boosted gene delivery, but you see these polyhedral shapes that perhaps led to a better interaction with the cells and better endosomal escape. So what we did next was we did multi-resolution imaging, in which we basically were able to look at individual lipid nanoparticles inside the cells and how they move. So this is a 3D rendition of an imaging experiment where the LNP basically is stuck and is bouncing back and forth of the endosomal compartment, whereas the ELNP has much more faster diffusivity in the cells. And once we quantified like 280 different images, we found like around 5-fold higher level of ELNPs were showing these larger diffusivity, which suggests either endosomal escape, but it also could suggest that they are entering into much more permissive environment of an endosome. Next, what we did was with a graduate student, Marco Herrera and David Kim, we used this galectin reporter that has been reported in the literature, where galectin-8 is a cytosolic protein. If there is an endosomal rupture, the cytosolic protein starts to bind on the surface of an endosome, and that causes endosomal escape. So it's a good reporter system that you can then image endosomal escape. So here is what we did. We made these galectin-8 based cell lines with GFP. You see these are for 24 hours. You see cytosolic appearance of the galectin-8. Once you add cholesterol, you see these rare events where you see these punctate structures that represents an escape. It's hard to visualize escape otherwise, because again, as you can see here, these are much more rare events. But once you add our ELNP or those containing cytosterol, you see these massive events. And once we quantified that this actually was the late endosome lysosomal environment where NPC1 is also present, where you are seeing much of this activity. But this gives us one way of looking into the escape process, because now you can image a lot of escape. So based on that, what we did next was we started to use the strategy for different sort of treatments and disorders. One of the primary thing was SARS-CoV-2. So when the pandemic hit in 2020, we thought we would like to take part in the effort against this disease. So Yu-Kyoung Kim, who's a graduate student of mine, and he's also a graduate from Yonggam University in South Korea. He basically for 3 months went to the lab alone. We had all the permissions and developed this strategy where we could use a soluble form of ACE2. So the SARS-CoV-2 basically binds to the receptor called ACE2. The ACE2 also has a soluble form which can neutralize SARS-CoV-2. So our idea was, if you can deliver the soluble form of ACE2, it can capture. If we developed an inhalation or an injection, the soluble form would be secreted by the cells and can capture SARS-CoV-2. To do this, we basically engineered a soluble ACE2 variant which didn't have the transmembrane domain, so that it can be secreted out in the extracellular milieu. Once we engineered that, we transfected the cells with ELNPs, and what you find is in lung culture cells, in the cell culture media, you find production of soluble ACE2, which is much more higher with ELNPs as compared to the regular LNPs. And then in the Western blots, also you see that the protein is being formed. What we did next was we did an intratracheal installation of the ELNPs with luciferase, and you see lung transfection there. Once you give the soluble ACE2, and then in the bronchoalveolar lavage, we started to see the appearance of the soluble ACE2, indicating that this could be used for neutralizing any SARS-CoV-2 that enters through the lungs. Next, we also performed, we made a pseudovirus for SARS-CoV-2, which has had a luciferase in it. And then we basically had overexpressed the ACE2 in cells, and what we find is that ACE2 is definitely overexpressed ACE2 is needed for SARS-CoV-2 basically function. And once you then transfect the soluble form of ACE2 in cells, the SARS-CoV-2, which has luciferase in it, it basically can go in untreated cell population. But once you treat them with soluble ACE2, you see this sort of inhibition as a positive control. The VSV pseudovirus, it affects cells independent of the presence or absence of the soluble form of ACE2. So currently, we are working with collaborators to show that this can also function against the live virus in animal models, and hopefully we'll have that coming out soon. Before COVID, we were also working in the area of cystic fibrosis, and that actually helped us to move into the lung space pretty quickly. CF is a fatal disease that is caused by the absence of CFTR gene that leads to a chloride secretion. Absence of this gene causes less chloride secretion and a mucus that thickens, and that leads to secondary infections, and that can be fatal in patients. Although there were some drugs that are now clinically approved, there are 1,700 known mutations. So if you could deliver an mRNA for CFTR, it is possible that you can treat different kinds of diseases altogether. So what we did here was we took a CFTR mRNA and then transfected the nose of a CFTR knockout mouse. So in CF patients, one of the key clinical sort of things that to figure out if someone has CF or not is a nasal potential difference, because the airway epithelium and the nasal epithelium are basically very similar. Through nasal potential differences, you could see if the chloride secretion is happening or not. So once you give back CFTR, what we found was recovery of the chloride secretion as evidenced by this nasal potential difference. And then what we did next was that rather than just going after CFTR, so CFTR basically is pumping chloride in, but absence of CFTR, there is another channel called ENAC that starts to overexpress, and that sends the sodium inside, and wherever sodium goes, water goes, and mucus becomes thicker. So a lot of people are trying to inhibit ENAC against for treatment of CFTR as well. So rather than using siRNA, we made a mutant form of one of the components of ENAC, the alpha ENAC mutant, which if the dominant negative form of ENAC is expressed in cells, it basically forms the trimers and inhibits the function. So rather than silencing or using siRNA, you can use dominant forms of RNA and block functions of proteins as well. So currently we are basically trying to get the CFTR mRNA and ENAC mRNA in one place and deliver them both together in the airways and see if they have a much better potential to become a therapy. And what we showed was that using just the ENAC transfection, we do see a decrease in the sodium dependent currents that are happening. And the hope is that, again, like if you are able to give an inhibitor using an mRNA-based approach, you can very easily combine CFTR mRNA with ENAC mRNA, and you'll express CFTR and inhibit ENaC, and we are excited to move that forward as well. So one thing we are also trying to do is to nebulize these particles. And one critical problem with lipid-based nanoparticles definitely is if you put them in vibrating mesh nebulizers, they tend to break apart, they aggregate. So you have to make a stable enough particle, but that particle should break apart once it gets inside the cells. So again, Jung-Won Kim in my lab has recently been able to make different formulations of these LNPs, and we find a pretty good transfection in the lung after inhalation of these LNPs with these formulations. And currently we are using, CFTR-based mRNA approaches and knockout cells and nebulizing the particles, and hopefully we can give the genes to different cell types in the lung as well. And finally, one of the other area of my lab, and which I'm really very excited about, is drug delivery to the back of the eye. So the eye is composed of, the retina in the eye is composed of different cell types. These are neuronal cells that are essential for channeling the visual input that is coming from outside into electric signals that go to the brain. So among these different cell types, the retinal pigmented epithelium and the photoreceptors are basically the key sites where, if mutations happen, that causes blindness. So RPE is epithelial cell lines, the photoreceptors are neuronal cell lines, and every day when we wake up, some of these photoreceptors are engulfed by the RPE and then are replaced. So if there is mutation in an RPE, then those phagocytosis doesn't happen, the photoreceptors tend to die, and this is, the photoreceptor is the key cell type that can cause, that is required to convert the light that is coming from this direction here to an electric impulse that goes to the brain. Similarly, there can be mutations in the photoreceptors, and that can do the same thing. There is a lot of photoreceptors that causes retinitis pigmentosa, and slowly, as it's a inherited disease, so children slowly start to become blind. And to halt this, there is one treatment using an AAV where there is an injection that helps that case, but again, AAVs are limited to one single injection and only for RNA or that is pretty small in size. So the challenge with the delivery to the eye is the best possible way to deliver to the back of the eye is to do a subretinal injection. It delivers directly to the retina. It is done surgically, but it requires like 6 different clinicians to be present. It is currently clinically approved for some of the therapies, but it is highly invasive. It detaches the retina and it has those limitations. The other possibility is to give an intravitreal injection, but in that case, what happens is that the material basically gets stuck in the aqueous environment, which is the vitreous humor, which is viscous in nature. So we need to find ways that we can deliver nanoparticles, especially that are having mRNA that can cross some of these barriers. This is one example that further corroborates what I'm saying. If we give a lipid nanoparticle that has an mRNA subretinally, you can see the retinal pigmented epithelium cells that they start to express the genes. But if you are giving directly in the vitreous cavity, a lot of the particles are not being able to cross these barriers, and you're seeing the transfection just happening in the outskirts of the retina, while you need to deliver your genes to the back in this area. So in this regard, with one of my collaborators, Dr. René Rayals, we basically tested a bunch of cationic lipids, ionizable lipids, and found that MC3-based lipids after subretinal injection do work for the RPE. We then used an AI9 mice and delivered a 3 recombinase, and you start to see editing because of that. And after a subretinal injection, again, you find if you change the PEG concentration and the cholesterol composition, we still find that the transfection is in the RPE, less so in the photoreceptors, where also there is a lot of mutations, and we need to deliver genetic cargo to the photoreceptors as well. if you give intravitreally, even after making these compositional changes, we see that a lot of material is sitting in the vitreous. So the potential after a subretinal injection, the potential mechanism by which the epithelial cells, those phagocytic epithelial cells are getting transfected is still sort of unknown. If you take an ApoE knockout mouse, you still see that an LNP that has mCherry in it is able to transfect the RPE, that's the back of the eye. MERTK, another phagocytic receptor in the eye, you still see that. And so currently we are working with a lot of my students to figure out, can we hit the photoreceptors and other cell types in the retina? And can we also develop ways that we can deliver nanoparticles through the intravitreal route, and they can cross these barriers and go back? And we have some progress there, and perhaps in like 3 or 4 months, you'll see a paper from us where we have shown some progress in delivery across these barriers and to different cell types in the eye, and we're very excited with that. So again, this is one example where now we have the capability of delivering molecules after an intravenous injections to the photoreceptors and other cell types in the eye, and not just the RPE. And that opens a lot of different avenues for using lipid-based nanoparticles for gene delivery and more likely gene editing to treat blindness. And I think that's something which can, we can start using sort of these materials in the extrahepatic targets as well. So finally, I think, we are in a very exciting time. After liver and delivery to endothelial cells and lungs that we have heard, I think localized delivery is also one way. There is examples of in utero delivery of the LNPs. We are working on the eye. You can give a direct injections to the brain to see some delivery. Short, I think, is another elegant way of using materials or polymers, like Dan Anderson was saying, or the vitamin compositions with Yizhou was saying for targeting the macrophages. So as we move forward, I think localized delivery or through intravenous delivery are starting to move beyond the hepatic and the immune cells towards the eye and towards the macrophages and things like that. And I think the advent of things like DNA barcodes would further make it possible that we can rapidly, using high throughput strategies, find which composition is the best one to move forward. And new material chemistry and unique ideas, I think, would definitely move the field further. And understanding the trafficking mechanisms as we find newer and better materials, I think, would be the key to make and evolve these materials to be even more potent and more directed towards a tissue and cell type towards the tissue as well. So I think the field is slowly starting to move there. And with this COVID pandemic, it is clear that this has a transformational impact in lives, and that is possibly going to happen in different other organ types and other diseases as well. So finally, I have great sets of grad students, postdocs to thank, current collaborators, CF Foundation, and our funding. And with that, I'm happy to answer any questions that you have.
So we have one question from the floor. So you showed the high efficacy of the cytosterol-based LNP delivery. So what makes LNP cyto provide a higher efficacy? Does it mean the higher cellular uptake or higher endosomal escape? What is your explanation about that?
Yeah, that's a great question. So like we showed before, so we think that's higher endosomal escape, because what you're seeing with cytosterol is we see those much more linear trajectories inside the cells. And using that galectin assay, which I was talking about, where the galectin, so that's like unlabeled particle, which you are throwing on cells. If the galectin 8 is an endosomal membrane, then that shows escape. So we are seeing a massive escape once you put even unlabeled ELNPs on these cell types. And so we also did and published that there is not much of a difference on the uptake. It's the endosomal escape, and that is corroborated by the cytosterol and the 3D multi-resolution imaging that we have done.
So have you ever compared the endosomal escape efficacy of the cytosterol-based nanoparticle as compared to the penetrating enhancing endosomal penetrating enhancing peptide-based LNP? Have you ever compared? Is it possible to compare that kind of efficacy?
Yeah, I think once you start adding peptides, these things start to behave a little more in a complicated way. So I think there were papers before where people have used cell-penetrating peptides, and what I can say is with the ELNPs, even if you are putting some peptides, the cytosterol basically is helping in breaking apart these materials in a better way. So then the ionizable lipid interacts with an endosomal membrane in a faster way, so to speak. If you add like a targeting ligand, then perhaps it is possible that it will enter some of the cell types in a better way, but the escape might be better due to the ability of the cytosterol for better, sort of the packaging differences that are happening in the system. So there are still, though, a lot of different peptides, endosomal escape peptides that one can try as well. But things get complicated once you start putting stuff on a nanoparticle. If you just replace it, it's just much more easier to control the particle and see if it works better in that system. But I think it's a possibility in the future where you can combine these two parameters as well.
Okay. And I have a question about the stability of LNP. So
Right.
how stable is LNP in vivo? Do you have any strategy to increase the in vivo stability of LNP in the future?
Yeah, I think people have try to use like LNPs that have, they've changed the peg or the peg length to improve the stability or circulation of LNPs. But what also ends up happening sometimes that you make a particle more stable and it'll stop working. We actually tried like making a super stable particle and then adding cytosterol in it, and we have some data where it really works well in the context of lung. But you start to lose the ability of the material to again disengage and becomes stable enough not to be active anymore. So there's a balance between maintaining the stability of the particle, but also then making sure that once it's inside the cells, it sort of decomplexes and leads to a better escape. So it can cut both ways, if you end up like making a super stable particle.
All right. And one question is, as you know, some people are studying now ocular mucus penetrating nanoparticle, but you used that kind of retinal injection. So what do you think about that kind of mucus penetrating nanoparticle for your gene delivery for retinal degeneration application?
So we have come up with our own strategy of penetrating using certain ligands. So again, like that's work in progress, and hopefully, in a few months we'll have that story out as well. But we have been able to find certain strategies that actually really work well to help in penetration and can cross some of these barriers. But there could be other strategies as well. So I think that's one of the strategies, but of improving intravitreal delivery of nanoparticles. So stay tuned for.
All right. And in your extrahepatic delivery strategy, you mentioned about DNA barcode. For some beginners in this field, can you explain briefly what is the concept of DNA barcode?
So, we're not like the leading lab there. I think James Dahlman is one of the leaders in the DNA barcoding, and I think Dan Anderson has with James have like initial papers on barcoding as well, where the idea is that within the lipid nanoparticle, you could basically, as you are making all these different LNPs, you are adding DNA sequences that have that acts like a barcode. So you have one LNP that has barcode X, the next LNP has barcode Y, and then you can mix all of them, pool them together, inject, and because they are DNA barcodes, you can do next generation sequencing to identify which X barcode reached to a certain tissue, and that means that a certain LNP helped enable that. Rather than testing one material, you're combining all these particles together with each nanoparticle with a unique barcode, and then using sequencing, identifying, which barcode reached to which cell type, and then from that, assessing that which lipid nanoparticle was successful in doing it. So by doing this, what you're doing is, again, making it simpler rather than injecting a lot of animals, a few animals, and you're injecting like 30 or 40 different particles and one shot in them, and identifying which particle reaches to which cell type. And I think that's a very powerful tool of screening. I think we'll still need better engineering and things like that and fundamental understanding, but as a brute force sort of screening technology, that's something which can pave way to extrahepatic delivery.
All right, so it's kind of taking a specific nanoparticle to trace the kind of in vivo or intracellular fate of the nanoparticle, sort of, yeah.
Yeah, it's basically taking a specific DNA and encapsulating it in a particle. And that DNA is sort of a tag for that particle where it reached.
All right, thank you so much. Very enlightening talk. So thank you, Sahay. I'll give you a big applause, although you cannot hear. So thank you so much. All right, so now I'll conclude the first session, and the second session will start after a 5-minute break. I appreciate all your attention for the first session, and please stay tuned.
Okay. Okay, everyone. We are going to start the second session. For the second session, Professor Yizhou will chair the session, and I'd like to give him a mic. Please start, Yizhou.
Yeah, thanks, Hyukjin. Welcome back. It's my great honor to chair this second session. I really enjoyed the first session. There were great talks, and also I'm really looking forward to the talks in the second session. Let me introduce our first speaker. If I pronounce your name or university incorrectly, please forgive me. So our first speaker is Professor Dongki Lee. He's in the Department of Chemistry, Songyong K1 University. He is currently serving as the Asian editor of the Nucleic Acid Therapeutics and editorial board member of Molecular Therapy Nucleic Acids. His work on siRNA structures, nucleic acid aptamers, and eukaryotic gene regulation has been published over 80 papers, including prestigious journals such as Nature, Cell, PNAS, and Molecular Therapy. In 2010, he founded OliX Pharmaceuticals, a publicly traded RNAi therapeutics company, and he serves Chief Executive Officer. Now let's welcome Professor Lee.
Thank you. Thank you so much, Dr. Ball. Can you see my slide?
Yeah.
Okay, good. Well, I'd like to start by thanking the organizers for invitation. It is very great honor to speak in this exciting conference. So as introduced, my laboratory in Songyong University is focusing on the RNAi mechanism of action, and we wanted to develop the real RNAi therapeutics, so that I founded Orix Pharmaceuticals in 2010 by transferring the RNAi technology from the academic laboratory to the company. So today, hopefully, I can show you some of the recent data from Orix regarding the various RNAi therapeutic programs, and also different delivery modalities. So I was asked to keep it short, so I'll just skip this slide. RNAi is considered the most powerful oligonucleotide technology. I have to briefly talk about the RNAi mechanism. So short double-stranded RNA called siRNA is introduced inside the cells, and they are associated with RISC. And one strand called guide strand remains in the RISC, and the other strand, the passenger strand, is removed. Now, the RISC guide strand complex will scan for target mRNA complementary to the guide strand, and when it finds one, it binds and it cleaves the mRNA off to inhibit the protein synthesis. And this process is catalytically repeated, so that the small amount of RISC-loaded siRNA can really perform excellent target gene silencing. So, so far, there are 4 RNA therapeutics approved, either FDA or EMA, all from Alnylam developed. I think most of the researchers, at least in academia, are using this form of siRNA, so-called conventional siRNA or tissue form, to trigger gene silencing, target gene silencing of their gene of interest. In my laboratory, we actually came up with a different version of siRNA called asiRNA or asymmetric siRNA. And in this asiRNA structure, you can see that the sense strand, the strand that is removed when associated with RISC, is shorter than the conventional siRNA to make it a short duplexed structure and also asymmetric structure. We can either maintain the length of the guide strand similar to the conventional siRNA, or we can make it even longer, such as 25, 26, or even 31 nucleotides long. So this asymmetric siRNA
we found that this actually is beneficial in several aspects. So first of all, it gives comparable level of gene silencing. But more importantly, we were able to observe that this asymmetric version of siRNA can reduce various non-specific effects, such as off-target effects or RNAi machinery saturation. And also, our collaborator, Dr. Jayakrishan Ambati, has published a series of papers to show that the conventional 19 base pair long double-stranded RNA triggers non-specific TLR3 reactivation when introduced inside the eye, whereas the shortened double-stranded RNA does not trigger this non-specific TLR3 reactivation. So this is great, but to be able to make this siRNA into real drug-like molecule, we need to first deliver the oligonucleotide inside the cells. So how to deliver siRNA inside the cell? As we all know, that delivery of siRNA to liver has been so far very successful. It started off by lipid nanoparticle, the topic for the most of the previous speakers. Patisiran, the first FDA-approved siRNA therapeutics, was delivered to the liver hepatocyte by LNP. After that, mainly developed by Dr. Manoharan's team at Enlighten, now we can effectively deliver siRNA by simple conjugate of sugar, carbohydrate, GalNAc, or N-acetyl galactosamine. So the second, third, and fourth, and also the coming up siRNA therapeutics are all based on the GalNAc conjugate and not using any delivery vehicles. So delivery to the liver is great, but we also want to deliver extrahepatic tissue. So local delivery, like skin, eye, or even CNS, the direct delivery of siRNA RNA to the diseased tissue is possible by simple lipid conjugate. But it's more challenging to deliver extrahepatic tissue in internal organs. So currently there are a number of efforts like peptide siRNA conjugate. And Lion actually yesterday made a collaboration agreement with Peptid Dream, a Japanese company focusing on peptide ligand development. Also, antibody siRNA conjugate is currently being pursued by a company named Avidity Biosciences. So hopefully these efforts can really expand our targetable organ to deliver therapeutic siRNAs to patients. So today I'm going to talk about the examples of our own examples by two delivery methods. The first one is by local delivery by lipid conjugate, and the second example is liver hepatocyte targeting by GalNAc conjugate. So we have developed siRNA platform called cell-penetrating siRNA. So here, the simple lipid conjugation at one end of the sense strand enables the whole siRNA molecule to enter cells without the aid of delivery vehicles. And this, and we started with cholesterol, and for skin delivery, we are using cholesterol. But for ocular and other tissues, actually, different lipids needs to be screened mainly because of the toxicity issue. So we have for various target genes and various cell lines and tissues, we see excellent in vitro and in vivo data. Our first program is ORX-101A, is targeting hepatocytic scar. And hepatocytic scar is produced during the wound healing process when the excessive fibrosis, uncontrolled fibrosis occurs. So we decided to target connective tissue growth factor, CTGF, which is well known as a master regulator of fibrosis. And this work has been published. So this is our CPA siRNA, where we have 16 nucleotide long sense strand and 31 nucleotide long antisense strand with cholesterol conjugate. And we were able to show that in a rat excision wound model, the intradermal injection of CPA siRNA effectively reduces the target gene expression, as well as other the fibrosis markers. And currently, this program is in undergoing phase 2 clinical trial in Korea, as well as in US. We then tackled on another skin disease called androgenic alopecia, or better well known as male baldness or male hair loss. So here we are targeting androgen receptor. So basically, the mechanism of male baldness, male hair loss, is the action of testosterone. So testosterone is modified and it enters inside the nucleus, and it activates androgen receptor to trigger various gene expression, which results in the hair loss process. So we decided to inhibit the expression of androgen receptor to block the hair loss process. So this result shows that intradermal injection, single injection of our CPA siRNA targeting androgen receptor can maintain the suppression of androgen receptor gene expression more than 50%, at least up to 3 weeks in mouse model. It also shows that this is actually the human explant of a hair follicle. And you can see that the androgen receptor expression is effectively suppressed by incubation of our CPA siRNA. To see the therapeutic efficacy in animal model, we set up the testosterone-based the hair regrowth model in mouse. So you shave the back of the mouse and you keep injecting dihydrotestosterone to suppress the regrowth of the hair. And whether we inject the control compound or our siRNA. And as you can see here, compared with the genetic control, the intradermal injection of CPA siRNA really boosts up the regrowth of the hair in the back of the mouse, and also is also verified in H&E stain. This is a quantification of hair weight. So we shave off the regrown hair and measure the weight, and you can see the significant regrowth of the hair, and also significant increase in anagen to telogen ratio. So basically, you go to the telogen cycle and you permanently lose the hair, and we are suppressing the anagen to telogen transition. So this is very exciting result, we believe, and we are currently initiating the IND-enabling study for clinical trial. We can also utilize this CPA siRNA delivery to deliver siRNA to the back of the eye. As I already told you, so we initially started with a cholesterol conjugated siRNA, and it turns out that somehow this cholesterol siRNA conjugate is very toxic to the retina. So we had to screen for less toxic and still maintaining the gene silencing potency, and we were able to come up with some of the alternative lipids. And this is one example of intravitreal injection from top to the bottom. So whether the fluorescent labeled siRNA goes all the way back to the back to the eye, all the way down to RPE, and you can see that after several hours, actually, these siRNAs successfully reach the RPE, and we have seen by testing several target genes expressed in RPE that we can effectively suppress the gene expression. So based on this delivery format, now we have two programs targeting A and B, ORLIX 301A and B. So A and B is a disease where the region called the macula in the center region of your retina gets degenerated and you lose vision. And there are two types of AMD, wet form and dry form. So wet AMD, there are several therapeutics. Basically, they're all anti-VEGF therapeutics. But unfortunately, the dry AMD, there's no currently available therapeutics. And also the subretinal fibrosis, which is the disease. Basically, you're getting scar in your retina because you keep getting injury and recovering and repeating this step, and you basically trigger fibrosis in your retina, which result in irreversible vision loss. So we decided to go after these two diseases where there's currently no available treatment. So this is one of the mouse dry AMD model, also developed by Dr. Amati in University of Virginia. And subretinal injection of ALU RNA, the non-coding RNA, can trigger retinal degeneration pattern, as you see here, which represents the late-stage dry AMD, so-called a GA or a geographic atrophy. And the intravitreal injection, our lead compound, can actually effectively suppress the retinal degeneration by fundus imaging, as well as optical coherent tomography, OCT imaging as well. This compound is also effective in wet AMD model. So this is actually a non-human primate model where we trigger, we irradiate the back of the eye of the monkey with laser, and we see the leakage area. So basically, we see upon intravitreal injection of our compound, we see a significant reduction of retinal leakage area, as well as a reduction in retinal thickness. And this effect was maintained at least up to 8 weeks or 56 days. And now we believe we should go further because we actually stopped the experiment at 8 weeks. Another program, ORIX-301B, is actually targeting same target, CTGF, as we go after in a skin scar program, because basically the fibrosis mechanism should be very similar. So we also found that this CTGF targeting siRNA can effectively trigger reduction of laser-induced blood leakage, as well as the collagen formation in mouse model. So both programs are currently in the middle of the GLP tox study, and we are planning to file IND for clinical trial in early next year. So now I'd like to change the delivery methodology to talk about liver targeting by GalNAc conjugate. So again, as I told you, the early effort by Dr. Manoharan really set up this whole delivery platform. So Dr. Manoharan's group at Atlanta has shown that the tri-antennatic GalNAc, the trimer of N-acetyl galactosamine sugar linked to the siRNA, can successfully effectively target hepatocyte, the membrane where the ASGPR receptor is present. So ASGPR receptor is a trimer, so you require, it is thought that you need at least 3 GalNAc molecules. And they bind and they internalize by receptor-mediated endocytosis, then trigger RNAi. So the Atlanta's original development of the tri-GalNAc is like this fork-like structure they call tri-antennatic GalNAc, but they've later shown that actually the trivalent GalNAc is also effective, and this was also verified by other groups. So at ORIX, we actually licensed in this linker chemistry from a US-based company named Chemical. So I'd like to just mention that the GalNAc molecule itself is patent-free because it has a long history. It's really how you link this GalNAc molecule to the RNA. So basically, the linker chemistry is where the IP is. So we set up our own GalNAc ASIRNA delivery platform. As you see here, like as you expected, GalNAc-based siRNA delivery is a well-established technology. So as you expect, you get very nice delivery to the liver and also rapid and effective uptake in mouse hepatocytes. And because the siRNA molecule is not protected by LNP or other complex, you have to chemically modify the siRNA. And we were able to find the most effective chemical modification, which maintains the maximal target gene silencing, as we see here. this chemical modification pattern can actually achieve more than 90% target gene knockdown, at least up to 50 days, which is comparable to other competitors' GANA platform. So with this, we initially start with the targeting HBV, hepatitis B virus, mainly because although we understand that there are other RNAi companies with clinical stage HBV program, we wanted to go after this anyway because first of all, we were able to find some sequence position which gives us the freedom to operate, to work with our own ASIRNA, and also the fact that most of the HBV patients are in Northeast Asia really so make us think that this is attractive target. And also because the other companies' data are already available, we were able to compare our GANA chemistry with our GALAC platform with other companies to see whether our GANA platform is really competitive. So just to show that this result is the AAV-based HBV mouse model. And you first inject adeno-associated virus-based HBV, then single subcutaneous injection of GANA siRNA. And we look at the S antigen, E antigen, and HBV DNA level, and all of them show at least 2 log, more than 100-fold reduction in plasma level, which is at least up to 28 days, which is, as I already told you, comparable, expected, and comparable with other companies' preclinical results. The second program we are currently focusing on is targeting NASH, which stands for non-alcoholic steatohepatitis. So basically, you get NASH because because of the Western diet, like hamburger, pizza, or whatever, and then obesity. And you start with fat liver, then you get inflammation in your liver tissue, which then undergoes like liver fibrosis, cirrhosis, and eventually cancer. And NASH is very attractive target, but especially we decided to go after this target because our scientific advisor from Harvard Medical School actually suggested one of the target based on the genome-wide association study, GWAS, which was already associated, thought to be associated with protection from NASH liver fibrosis in humans genetic studies. So we set up high-fat diet model in-house, and we do a couple of injections, and we look at all the markers. First of all, target gene knockdown is achieved at least up to 90% or even further. And very interesting and surprisingly, we were able to see a quick, rapid loss of body weight up to actually 13 points or something, so which was very interesting. We then look at the histology of the liver tissue. This is normal chow, so this is what the normal liver should look like. And this is a NASH mouse liver. You see all these lipid vacuoles, droplets. And our siRNA injected mouse actually, you see very striking difference between the NASH mouse, high-fat diet mouse, almost to the level of a normal chow mouse in histological analysis, which is also seen in this oil red staining. So oil red staining basically stains the lipid droplets, and as expected, based on the previous slide, that we also see significant reduction of oil red staining in our siRNA injected mouse. And also we see all the favorable changes in liver function, AST/ALT, and total cholesterol, triglyceride, and LDL levels. So we believe this compound is actually excellent, a promising candidate for NASH therapeutic development. This slide just summarizes the ongoing therapeutic program in OLIS. And I'd like to just finish by just briefly mentioning the challenges. So it's great that we are able to target liver, but we are only targeting hepatocytes. Still, there has been any meaningful clinical results targeting, for example, hepatic stellate cells, which is the main target if we want to go after liver fibrosis. Extrahepatic tissue. So I told you about the example of local delivery like skin and eye, but other internal organs, we still need to develop effective delivery technology to be able to go after more life-threatening and serious disease indications using siRNA and other oligotherapeutics. So that's all I have prepared. Thank you for your attention. And if you have any questions, I'll be happy to answer. Thank you.
Hyukjin, have you got any questions from the audience? By the way, thanks, Professor Lee, for the wonderful talk.
Thank you.
Yes, we are collecting some questions from our audience. Yes, there are a couple of them. First of all, what is the mechanism of asymmetric siRNA? Is it similar to regular double-strand RNA that directly targets the AGO proteins? That's the first one.
Yes, it is essentially identical in terms of the RNAi triggering mechanism, identical to the conventional siRNA. We just reduce the length of the sense strand a little bit to be able to alleviate some of the side effects. So just want to mention that if you keep reducing this length of the sense strand, actually you start to drastically lose the efficacy, presumably because you need certain minimal double duplex length for risk AGO2 complex to capture the double-stranded siRNA.
Okay, there is a second one. For ocular delivery of siRNA, is there any delivery platform that your team is using?
Yes, as I already told you, so we are using lipid-based conjugation. At this point, unfortunately, we are not releasing the formulation of lipid, but I can tell you, as I already told you, that we initially used the cholesterol, and for skin delivery, cholesterol works beautifully with no little side effect. But ocular and also other tissue, like pulmonary and other tissues, these tissues in general seems to be more delicate, and they are more susceptible to local inflammation. So we have to screen for the lipids which is most acceptable for each specific target tissues.
Okay, this will be the last question. So how long do you think your mRNA knockdown will rest for AMD treatment for the single injection? And is there any other way to improve that you do the less frequent injection to your eye?
Based on our non-human primate result, we have seen that at least the efficacy lasts at least a couple of months, and we are positive that we should be able to perform like a once in every quarter treatment in actual clinical trial. The duration of siRNA efficacy really depends on the tissue you are targeting. So we are not sure how long we could maintain the efficacy duration in the retina, but I understand that there are some like a polymer or some engineering-based method where you inject some of this implant inside your eye and then trigger slow release of the drug ingredient. That could be one of the options in the future.
Thank you very much.
Thank you.
Yizhou, it's your turn.
Great. Let's thank Professor Lee again. I would like to introduce our next speaker, Professor Dao-Hee Min. She is currently a professor at Seoul National University and the co-founder and the CTO of LemonX. Professor Min's research focuses on the development of drug delivery systems especially for functional RNA, DNA, and the protein, with a collective understanding on nanosurface chemistry and the diverse biomedical applications of porous nanomaterials. She received various prestigious awards, including KCS Wiley Young Scientists Award, Korea L'Oreal UNESCO Fellowship Award for Women in Science, and the Order of Science and Technological Merit Doyak Medal from Korea government. Let's welcome Professor Ming.
All right. Thank you for a nice introduction, Professor Dong. It's my great honor to give a talk today, and I'd like to thank all the organizers and speakers who gave great presentations, Professors Anderson and Dong, Sahay, and also the Professor Siegwart. Okay, let me talk about RNA therapeutics based on porous solid nanoparticle degradable. So here, I'd like to present a totally different concept of drug delivery system from LNP, because my concept for the drug delivery system for RNA therapeutics is based on like solid porous nanoparticles. It's not lipid-based delivery system. So let me start the presentation. So first, I'd like to give an overview about RNA therapeutics and also available drug delivery systems and what kind of aspects we should consider for the successful RNA therapeutics development. And next, I will talk about properties and basics about DegradaBALL, which is a porous inorganic solid nanoparticle system, and then principles and mechanisms of DegradaBALL-based drug delivery systems. And next, I will talk about case studies for the delivery and applications based on RNA for the therapeutics, and also plasmid DNA and proteins delivery based on DegradaBALL. So when we think about RNA therapeutics, there are three aspects we should consider. First, RNA stability, transfection capability, and also translation in case of IVT mRNAs, and also the targeting availability of RNAs in case of microRNAs and siRNAs. So when we talk about RNA stability, people use different strategies to improve RNA stability, because RNA molecule itself is very fragile molecule. So we need some kind of protection strategy to improve the half-life of RNAs in the body. So many people use chemical modifications to the RNA strands and structure modifications like 5' capping and poly-A tail, and also delivery vehicles could help enhance RNA stability. And also in case of transfection capability, nanoparticle strategies like lipid nanoparticles and also cationic polymers or solid nanoparticles could be applied. And for the translation efficiency, like modifications to the RNA structures could be one important strategy for good, efficient translation. So when we talk about RNA therapeutics, there are three popular APIs available, siRNAs and mRNAs. Let me first talk about short RNAs, including siRNA and mRNA. And these two different types of short RNAs share similar mechanisms inside the cells. So basically, they are used to suppress or knock down the expression of mRNAs sequence specifically. And in case of siRNAs, the mRNA cleavage is very sequence specific and it's a target specific. Whereas microRNAs could knock down multiple mRNA expression using just one single type of sequences. So there are slight differences, but both of molecules are popularly used for the knockdown of gene expression. And next, RNA therapeutics is IVT, in vitro transcribed mRNAs. And nowadays, it is very popular for the COVID-19 vaccine. And these mRNAs could be also used for the protein replacement therapeutics. So when these mRNAs are synthesized in vitro and is introduced into cell cytoplasm, then the ribosome could be complex to the mRNAs and the translation begin, and the wanted proteins or epitope or antigens could be like expressed. So basically, all these APIs are based on RNAs. So in case of RNAs, it is very difficult to achieve very effective intracellular delivery because RNA molecule is easy to be degraded by the RNAs, which is a molecule, which is like degradation enzymes, which are abundant in the body. And also the RNA itself cannot penetrate the cell membrane. So we need some the assist from the drug delivery system for the efficient intracellular delivery. So the DDS vehicles could be used for the protection of mRNAs and efficient delivery to into the cells. And here, the importance and also the success of RNA therapeutics could be largely dependent on the delivery systems. So people say the three important aspects of RNA therapeutics are first, delivery and delivery. So many like current delivery systems could share this kind of limitations, and we are like constantly trying to like improve or overcome these limitations, like unstable structure. So sometimes like lipid nanoparticles could fuse together over time, so we could not store that for a long time at room temperature. So we need to freeze down, and the transportation should be done at extremely low temperature also. And also the formulation process could be complicated, and drug loading efficiency is also not satisfactory. And sometimes the size distribution is hard to manage, and also sometimes we need to achieve sustained release of APIs, but some drug delivery systems only allow burst release, so the frequent injection is needed in that case. So when everybody is looking after like LNP-based drug delivery system, I sit back and thought about like completely different approaches for this RNA therapeutics. What if we can make some kind of a solid nanoparticles which can be stored at room temperature and also very stable at room temperature and stored at as a like powder form, and then we can load the APIs or RNAs inside pores into these porous nanoparticles. Then the formulation could be very easy, and the structure like stability could be very much like effectively improved, and also it could allow like a sustained release and durable drug efficacy, so that we can reduce down the number of injections and also require the dose for the drug efficacy. So that's how we developed this porous silica-based nanoparticles, so-called degradable. This you're looking at this TEM images of degradable. And in this, if you look at this TEM image, there are many empty spaces inside this particle. So in these empty pores, we can load various APIs, and we can perform like surface chemistries very flexibly to the inner surface of degradable and outer surface of the degradable. So that could give you very good variable flexibilities for the development of degradables for different target cells or different target organs. And also, most importantly, it is very stable under standard ambient conditions when we store it as a solid powder form. So it could be stable for more than a year at room temperature under standard conditions. All right, so based on this degradable system, we are developing we are developing different drug development programs based on siRNA, mRNA, and also plasmid DNA and proteins. And today I will focus on the RNA delivery systems and drug development programs. In case of RNA medicine based on degradable, we observed that it could allow efficient intracellular delivery and very good protection against RNAs, so it could increase the integrity duration of RNAs, and also it exhibited durable high efficacy with lower and fewer doses. So what is silica? Silica actually is a very popular component. Silica is generally recognized as safe, GRAS, by US FDA. And also, silica is often used in tablet form drug formulation as an excipient, and also frequently taken as a dietary supplement for nourishing hair, skin, and nails. And also, silica-based nanoparticles are already entered into clinical trials in USA. So when you type a silica dietary supplement at the Amazon website, you can find a lot of dietary supplement based on silica. So the silica is a component which are needed for the human health, and especially for the healthy connective tissue and hair and nails. So now I'd like to talk about drug loading, endocytosis, and release of APIs based on degradable. The loading is very simple. Just mix APIs with degradables in aqueous solution together, then you can achieve the complexation. And just handshaking and just incubation for several minutes at room temperature would allow about 100% API loading. And then when it's treated, it goes into cells by endocytosis. And then in the cytoplasm, the RNAs could be released from the degradable. And degradable is also biodegradable, as the name stands for. So it could be that safety and biocompatibility would not be an issue. So first, we investigated the protective effect of degradable of loaded cargo. So first, we used siRNAs as an API and loaded into ball, and then we treated RNAs in order to observe the protective effect of degradable for the siRNA integrity maintenance. So here we found that even after a treatment of a high concentration of RNAs to the siRNA complex of ball, we found that siRNAs stay intact even after incubation. And also that tells you the siRNAs, which are complex with ball, could give you very good knockdown effect in vivo. So on the right side, you can see that the very good knockdown effect of siRNA when it is complexed with ball. So basically, the good protection against RNase-mediated degradation would be a key for the successful RNA-based therapeutics. Next, we investigated cellular uptake and endosomal escape. First, we observed that how effective the cellular uptake of degradable into cells. And on the left side, we are presenting the bio-TEM image of degradable treated cells. You can see that many degradable particles are internalized into cells, and also some particles are about to enter into cells. And also on the right side, you are looking at the fluorescent images of cells which are treated with dye-labeled degradable, and you can see the red fluorescence, which is corresponding to the degradable, are well observed inside cytoplasm, not in the nucleus. So we confirm that this nanoparticle readily go into cell cytoplasm, but not into nucleus. And next, we assessed the endosomal escape property. And before we perform endosomal escape experiment, let me talk about endocytosis mechanism. So we wanted to reveal the mechanism of endocytosis of degradable. So we treated different like endocytosis inhibitors, and we found that chloropromazine inhibited the most like inhibited the endocytosis of degradable, suggesting that the endocytosis of degradable is mostly mediated by the class 3 mediated endocytosis. And also we next investigated endosomal escape. And here we stained the acidic intracellular compartment by using lysotracker dye. And then the dye-labeled ball was treated to the cells. And we followed the co-localization of the fluorescence corresponding to the lysotracker and ball. And we found that Pearson's coefficient decreased over time. And you can see that it decreased over time from 1 to about like 0.3 or 0.2 within 2 hours, which means that ball could be very efficiently escaped from late endosomes or lysosomes. And next, we investigated how efficiently the cargoes or payloads could be released in the cytoplasm from degradable. So on the left side, you are looking at the fluorescent images of cells treated with green fluorescent dye-labeled ball and red fluorescent dye-labeled siRNA. And we followed the fluorescence comes from these two entities over time. And we found that the co-localization coefficient decreased over time here, so which means that the cargoes, the siRNAs in this case, could be very efficiently and sustainably released from the ball inside the cytoplasm. And then we tested the durability and the efficacy of the degradable-based therapeutics. So we found that the efficacy is much more durable compared to LNP-based delivery of siRNA. So on the left side, the graph, you are looking at the comparison between the degradable-mediated delivery and LNP-mediated delivery over time. So you can see that ball could maintain the knockdown effect and efficacy over a long time, more than like 96 hours. But LNP-based delivery is causing the recovery of the target gene expression very quickly. So that means the degradables could achieve the sustainable release and durable effect of siRNAs inside the body. And also, this durable high efficacy was observed in vivo also. So here we injected Cy5 labeled siRNA, loaded Physi labeled degradable into tumor. So we observed that the Cy5 signal comes from the siRNA lasted much longer compared to just siRNA only injected the case. So it's like the long-lasting effect was observed over like more than 5 days actually. So that tells you that if you use degradable as a drug delivery system, then less frequent injection is like achievable and satisfactory for the same drug efficacy or even higher drug efficacy. And also at the same time, we could use just a very smaller amount of siRNAs compared to other strategies. For example, we observed that only like 100 times less amount of siRNA could be effective, equally effective compared to other drug delivery systems. And also long-lasting drug efficacy was also observed in case of protein delivery. So in this case, we loaded antibody, which is used for the immune checkpoint inhibitors in immuno-oncology area. And we observed that how long the the antibody signals could be observed after injection. And we found that the only antibody injected case, the antibodies diffuse throughout body very quickly. However, the ball could maintain very long-lasting drug molecule signal at the injection site. So that tells you also the local, very effective local delivery could be achievable based on degradable. So far I talked about the RNA therapeutics and drug delivery systems and introduction about porous solid nanoparticle-based RNA delivery system and the mechanism of degradable-based intracellular delivery of RNAs. So from now on, let me talk about different applications of degradables for the drug development program. So let me first focus on the RNA-based therapeutics. So as I previously already shown, for the RNA delivery with degradable, we just mix RNA with degradable together in a solution without using any ethanol or other organic solvent, just buffer or just the aqueous solvent would be sufficient. And after a couple of minutes of incubation at room temperature, it will give you the RNA-loaded degradables inside the pores. And these loaded RNAs could be protected against RNase-mediated degradation. And then this RNA degradable complex could be efficiently endocytosed, and then inside cytoplasm, these RNAs could be sustainably released so that we can achieve higher drug efficacy and less systemic adverse effects. All right, so first we tested siRNA sequence which could reduce the tumor size. In this case, we used siRNA against VEGF growth factor, and we found that when this VEGF was injected with the degradable, the tumor volume increase was well suppressed over time compared to other controls. And also, we quantitatively measured the efficient knockdown of mother gene, GFP, and VEGF genes in vitro using siRNA sequence. And you can see that on the left side figure, you can see that ball-mediated delivery of GFP effectively knocked down the GFP target gene expression. And compared to LNP, it was like 3 times higher efficacy in terms of knockdown efficacy. And also, it was interesting to find that degradable mediated delivery was very effective even in the presence of serum. It is important to observe this kind of data because in the body there are abundant serums, so we need some kind of very robust drug delivery system which could be effective even in the presence of abundant serum proteins. So here in vitro cell-based assay, we realized that this kind of properties of degradable mediated siRNA delivery. And also, we found that VEGF knockdown effect is concentration dependent, and also the efficacy, knockdown efficacy of ball-mediated delivery was like 2 times or 2.5 times effective compared to LNP-mediated siRNA delivery. And also in Vivo, when we tested the siRNA against GFP with degradable, the knockdown effect was observed, very good knockdown effect. And also VEGF knockdown was observed like here in the figure D. And this knockdown efficacy was much more higher compared to the LNP-based delivery. And also, in the xenograft cancer model system, we found that cancer growth was well inhibited when the siVEGF was injected in the presence of ball compared to other controls. And next, let me talk about different the RNAi based therapeutics for different indications here, like skin fibrosis. So the skin fibrosis, such as hypertrophic scar, frequently occurs in wound or surgical resections, and there are growing needs for effective antifibrotic therapies because current therapeutics in clinics are not very effective to treat this skin fibrosis. And also, fibrosis could occur in different organs also. So the anti-fibrosis drug development is a very important area. So to treat skin fibrosis, we targeted the CTGF, and we designed our own siRNA sequence to knock down CTGF gene, which is responsible for like fibrosis in many different organs, including skin. And when we treated this CTGF loaded degradable into cells, we found that also the siRNA could be sustainably released inside the cytoplasm, suggesting that it is like the sustainable and durable knockdown of CTGF could be achieved in the cells. And next, we compared this system with the LNP, and we found that the knockdown effect lasted much longer compared to LNP, as you can see on the left side graph. And also the downstream gene collagen 1 and collagen 3, which are directly responsible for the skin fibrosis, could be also downregulated when we treated LMS401. So that means that this LMS401 could be potentially very effective therapeutic options to treat skin fibrosis. And also we found that in the mouse wound model, we could recover the skin thickness and skin bumpiness. So skin smoothness could be improved a lot, and it was almost similar to the normal skin compared to the other controls. So we are very encouraged by this effective preclinical result. And let me summarize the LMS401 story. So based on this encouraging a knockdown of target gene CTGF to treat skin fibrosis, we are moving towards clinical trials, and we are expecting very impressive results soon. And next, mRNA delivery. So based on degradables, we recently found that it is realizable, and actually the mRNA delivery based on degradable was very effective and sustainable. So compared to the other type of drug delivery systems existing like DDS, the degradable showed that much higher and long-lasting the mRNA expression. In this case, we used the mRNA encoding luciferase, and after injection, we found that the expression lasted for more than 10 days after single injection. So similar long-lasting, higher expression of luciferase gene was also observed after intratumoral injection. So which means that we could use this degradable for the mRNA delivery to achieve effective vaccines to combat against infectious diseases, and also as a therapeutic vaccine against cancers. So first, we used a model plasmid DNA which encode GFP and luciferase. So as you can see in this image and the graph, the plasmid DNA expression was very impressively high when this degradable mediated delivery was used. And also this in vivo expression, plasmid DNA expression experiment also gave us a very good long-lasting, like high expression of introduced plasmid DNA. And in this case, we used the therapeutic gene, which could like improve the like wound healing. So in this case, we used the mouse model, the wound healing mouse model, and we found that wound closure and wound healing was like 2 times faster compared to control. And this is very interesting story. So far I talked about like nucleic acid delivery, including siRNA, like mRNA, and plasmid DNA. Now, these degradables could be also applied for the functional protein intracellular delivery. So here in this example, we used very large protein complex, which is called proteasome. And proteasome is responsible for the degradation of the like overexpressed proteins inside cells or ubiquitinated protein degradation. So this is a very large protein complex. So if we can like deliver these proteasomes into cell cytoplasm, then we can use that as some kind of therapeutics to treat like neurodegenerative disease, which could be caused by like overexpressed like abnormal protein overexpression. So here we used the proteasome to degrade tau proteins, which is responsible for like Alzheimer's disease progression or Huntington's disease. So when we treated the proteasome loaded degradable to the tau expressing cells, we found that the tau protein expression was largely like reduced after treatment of proteasome with degradable. And also that could be like the similar trend was observed in the quantitative immunoblotting experiment. And next, I'd like to show the therapeutic protein delivery by using Cas9 RNP to knock out myostatin gene. So here you are looking at the mouse behavior, like wire hanging test after injection of degradable loaded with CRISPR-Cas9 RNP, which targets the myostatin gene. And you can see that already the control mice fell down, but the drug-injected mouse could like hang on the wire much longer time. So twice longer time this mouse could hang on the wire. So which means the muscle strength was dramatically improved after knocking out this myostatin gene by using Cas9 RNP with degradable. All right, so as a summary, I introduced a very new concept of drug delivery system for the RNA therapeutics, which is degradable, based on inorganic porous nanoparticles. And this into pores, we can load various types of drug modalities. And in Lemonex, which I co-founded and serve as a CTO, we are running various drug development programs based on degradables, like siRNA therapies to treat skin fibrosis, mRNA therapeutics to treat infectious diseases and solid tumors, and protein therapeutics to treat cancers. So I hope in the near future we can see really like applicable drugs which could be used to help patients in clinic. Thank you very much.
Okay, so we actually received some questions from our audience. There are a couple. First one, how long does it take to dissolve mesoporous liquid particle inside of the cells?
So that's a good question. So dissolve meaning, I think dissolve meaning the degradation, right? So it is degradable inside the cells and inside the body. Like actually, the various factors and characteristics of nanoparticles could affect to the degradation kinetics. So In this case, we optimized the degradation kinetics so that we can achieve complete degradation of degradables within 2 or 3 weeks inside the body.
Okay. The other question was how can you actually control the pore size of these particles, because you're trying to encapsulate macromolecules inside of them, right?
Right. So the pore size control is very important because, like, physically the pore size should be slightly bigger than the cargos for the efficient loading. So during the synthesis of degradables, we are using some different conditions so that we can control the pore size.
Okay. And this could be our last question. What would be the in vivo fate of degradable? Is it really depend on the particle size or the surface character?
Actually, both like properties are like affecting to the like fate, and also the targeting and degradation and excretion. So excretion is like almost complete excretion of injected degradables was observed within about a month, actually.
And actually, we got one more question. So this is probably the last one. What is the mechanism for long-term expression of mRNA that is inside your silica particles? You told us that it's actually showing longer duration.
Right. So compared to controls, it was very amazing to observe the kind of very long expression of mRNAs. So we are still investigating exact mechanism of such a long expression. So I imagine that the pore size and also surface chemistry would be critical for achieving like long expression.
Okay.
Yeah.
So we're actually getting some more questions, but due to the time limits, I think Professor Min will answer it separately. Yeah, thank you.
Okay, thank you.
Okay, very nice work on the DegradaBALL. And we have our last speaker and great friend, Professor Hyukjin Lee. Hyukjin is a young Ewha Fellow Professor of Graduate School of Pharmaceutical Sciences and the College of Pharmacy at Ewha Women University. His research group focuses on the development of RNA therapeutics based on nucleic acid nanotechnology, particularly enzymatic and biological preparation of large-scale synthesis of RNA nanostructures for gene therapy. In addition, screening novel ionizable lipids and optimizing lipid nanoparticle formulation for targeted in vivo delivery have been a major research topic in his group. Hyukjin is also a wonderful organizer for this symposium, and he spent a lot of his efforts for preparing the itinerary. Thanks a lot. And Professor Lee, please.
Okay, thank you. It is my honor to actually organize and also having all the speakers from US and Korea. I'm very happy to share some recent progress of my work. Okay, some background of and significance. So as you probably know, we started with development of chemical drugs to proteins, and now we are having generic drugs in the market. As you can see here, genetic drugs usually by means of silencing protein expression, you use siRNA or microRNA, and if you want to express certain proteins, then you use plasmid DNA or messenger RNA. However, these nucleic acid drugs tend to degrade very fast when they are administered to the body. So usually there are many needs for the formulation of this nucleic acid drug, and in order to do it, you have to make various nanoparticles and administer to the local or the systemic injection, and you have to hit the right type of cells to do any kind of therapeutic effect. And now we are very excited with synthetic mRNA because it's well known for the very good expression of target proteins. By comparing with conventional plasmid DNA, their intracellular trafficking is a little different because plasmid DNA, they need a transfer of these large plasmid to the nucleus membrane. And then it will be transcribed and then translated to the proteins. However, when you use a synthetic messenger RNA, you only need to deliver those to the cytoplasm. So when you actually compare the transfection efficiency as well as expression, it will be dramatically different. As you can see in the below, by comparing plasmid DNA with mRNA, mRNA expression is far more potent. Also, the expression profile is quite different because plasmid DNA, it takes quite a time to get transcribed and then expressed. However, when we deliver synthetic mRNA inside of the cells, as you can see from the right, the expression profile is very rapid. You can actually observe the expression as early as 3 hours, and usually expression profile maximize around 16 to 18 hours. And depending on what protein you expressed, the half-life, depending on the half-life of the proteins, your decaying expression will be seen. So to deliver this mRNA, we use a microfluidic device to formulate. It's usually named as a rapid mixing. And the mechanism of LNP delivery to the cells usually depend on a receptor-mediated uptake. And in many cases, when those LNP are administered to the body, serum proteins called ApoE get absorbed on the surface, and those ApoE absorbed LNP interact with LDL receptors. Once they are internalized, those ionizable lipids do their job to disrupt the endosomal membrane, and the cargo materials such as mRNA can be delivered directly to the cytoplasm. So when we look at the components of lipid nanoparticles, previous speakers already mentioned a lot. There are 4 main components, including cholesterol, packed lipid, structural or helper lipid, and ionizable lipid. And the nucleic acid drug, you can actually encapsulate small interfering RNA, messenger RNA, or DNA as a choice of yours. And as you can see here, usually those structural lipid as well as ionizable lipid first interact with the anionic nucleic acid drugs to have electrostatic interactions, and cholesterol actually helps to maintain the structure as well as the packed lipid. And ionizable lipid, we already heard a lot of it, but has a very important function because these materials are the key to actually having electrostatic interactions during the formulation process. So as you can see here, a lot of amino alcohol candidates been used as a potent ionizable lipid. When they are incubated at lower pH, they actually do express strong positive character. So we do the formulation at lower pH to induce the electrostatic interaction between ionizable lipid and messenger RNA. After the formulation, we actually change the buffer to neutral to actually reduce the charge interaction and making LNP as neutral as possible. Once they are injected to the body, because of their neutral charge, unwanted non-specific interaction can be minimized. So LNP can have their stability. And when they hit the right target cells, they will enter as an endocytosis pathway, and when they are reside in the endosome, then lower pH will cause ionizable lipid to show a positive charge again, and will help the disruption of endosomal membrane. And as a result, you can actually deliver your cargo material to the cytoplasm. So in my lab, we used a lot of amine candidate, simple one-step reaction to make a ionizable lipid candidate and formulate them with siRNA or mRNA. And when they are formulated, they look like there is no multilamellar structure inside of them. So we think that it's just solid lipid nanoparticle. And they are injected directly to the mouse by IV injections. And we evaluate the efficacy of this LNP candidate by looking at an expression of firefly luciferase mRNA. And when we do the ex vivo image, mainly goes to the liver. But for just delivering firefly luciferase mRNA, we cannot tell what type of cells in the liver actually express luciferase. And the systemic delivery of LNP usually hits the liver tissue. And as you can see here, liver consists of mainly 4 different types of cells. Ones that are close to the blood is called sinusoidal endothelial cells, and there are Kupffer, stellar, and hepatocytes. And hepatocytes takes about 70 to 80% volume of the liver. And depending on what target disease you are tackle, you can either target hepatocyte and liver sinusoidal endothelial cells, or stellar, or Kupffer. And in my laboratory, we are interested in working on delivery to the hepatocyte and LSAC. However, as I already mentioned, just using a luciferase mRNA is hard to tell, which type of cells actually do express those proteins. So we actually searched previous studies on, consideration of a particle size for the delivery to the hepatocyte. And I think this paper was published by Peter Collis group. So there is small holes in LSAC membranes, blood vessels, and it's about size of 100 nanometers. So you want to make sure your particle is smaller than 100 nanometer to actually deliver those particles to the hepatocyte. So what we did, we actually changed a couple parameters in our LNP formulation, mainly the amount of PEG, and then deliver factor 7 and factor 8 siRNA together. As you can see here, when we increase the amount of PEG content, that means you are adding more surfactant that reduces your surface tension, so making your particle smaller, right? However, when you add too much of the PEG, your protein absorption under your LNP will be decreased, right? So a lot of PEG means that your chance to getting inside of the cell will be reduced. So as you can see here, you can easily see the U type of efficacy profile. So for factor 7, it's been used a lot for the monitoring of hepatocyte-specific delivery, and 1.5% PEG content actually showed the best result. For the factor 8 is marker for the LSAC delivery, and we get around 1%. So when we look at this profile, it will be quite difficult for us to actually differentiate the delivery to the LSAC and hepatocyte, because seems 1 and 1.5% works pretty well for both of the cases. So what we did, we try use a LoxP model, and we actually checked what kind of profile we get. So with PEG content of 1.5, that's what we get. Around 80% of hepatocytes been expressing TD tomato proteins, and LSAC around 40%. So you cannot control. So what we did, we actually increased the amount of PEG content to 3% to reduce the interaction between ApoE and LDL receptors. And then we incorporated mannose ligand to target the LSAC and CD206, I think, 206. So when we incorporate a mannose on our LNP, the enhanced uptake was observed, as well as it was very specific, because when we treated cells with anti-CD206, the increase of dose uptake was reversed. And when we tried enantiomer of mannose, the galactose, in this case, it didn't show any kind of reversal effect. So we thought that mannose to CD206 interaction is very specific, and it could help the delivery of those LNP specifically to the LSAC. So we redid our study. There is some breakage of the image, but what's going to tell you about this slide, when we increase the amount of pack content to 3%, we can actually reduce the uptake to the LSAC. And then when we used mannose ligand and we add about 2.5% of mannose pack to the system, we can direct LSAC specific delivery. And those immunohistochemistry image actually shows that only the LSAC actually do express the TD tomato. So this is our whole scheme. So we can actually control the delivery to the hepatocyte and also delivery to the LSAC by incorporating a ligand. Another short story for immunotherapy using a messenger RNA. As we all know that Moderna and BioNTech was very successful developing COVID mRNA vaccine. And for mRNA vaccine, it's very important to actually induce both of immune responses, like a humoral and cellular immune response. And for the cellular immune response, it is very important to actually generate a mature antigen-producing cells from dendritic cells. So it is still a discussion of using either unmodified ones or modified ones, but the expression profile is usually a lot better using modified mRNA. But as you guys probably know, the unmodified mRNA can also act as a protein producer, but also as a self-adjuvant materials. So there are still a lot of discussion, what should be used for the immunotherapy. But in my laboratory, we decided to actually use pseudouridine modified mRNA for the expression of antigens, and we actually screened a lot of adjuvant to go with, and we found that one adjuvant worked very well. And in our cases, it's called PAM3CSK4. It's a lipopeptide, and it can be incorporated very easily during a formulation of our LNP. And once they are incorporated, this is our scheme. So we will actually trigger the toll-like receptor 1-2, not 7-8, that is generally triggered by a single-strand mRNA. And as you can see here, we can just use a regular mixer to formulate, and the size was about same with the PAM incorporated LNP as compared to the regular LNP. And when we use luciferase to check whether this LNP can express after the IM injection, both of the LNP without adjuvant actually show very good expression. And initial immune triggering effect was evaluated by looking at TNF and interleukin expression. And we managed to actually evaluate the production of neutralizing antibodies, and as you can see on the top picture, the bar graph, both of the LNP actually show very good expression of IgGs. And for the cellular immune response, we actually need to make a CD8 positive T cells. So we actually evaluated the percent population of CD8 positive cells in this case. And as you can see here, the LNP without adjuvant was about 10%. And when we incorporated adjuvant, it was about 14.8%. 9%. So there are small increase. However, when we actually did a tumor challenge, as you can see from the growth of tumor size, with LNP, it managed to actually decrease the growth of tumor size as compared to the PBS and mRNA alone injections. But with adjuvant, it actually showed, best effect out of tested samples. Also, the survival was very good. Okay, summary. So synthetic mRNA is actually very good platform for rapid expression of target proteins as compared to the conventional DNA vectors. And so far, the lipid nanoparticles seem to work very well for both of the IV and local injections. And potential applications, previous speakers already mentioned a lot of these applications. So total therapeutic protein replacement. The other one is vaccine applications. And I think the third one is very interesting, like genome editing and cell engineering. So I think that's going to be the future of this technology. Thank you very much.
Wonderful talk. I got some question from chat. So what do you think about the best clinically used LNP for mRNA vaccine and siRNA delivery?
So first of all, mRNA vaccines, I think for the storage use, or longer storage, or the evading a cold chain, there will be a LNPs that is more suitable. But for vaccine applications, you have to think about what you actually want to achieve. Is it like you're trying to achieve the good expression of antigens, or you're trying to trigger more cell-based immune response? So depending on what you're trying to achieve, it will be different. For siRNA, I think the LNP applications only for so far is a partition and targeting for liver. And as you probably all know, now main pipeline is actually a conjugate. So GalNAc RNA conjugate, and that's more easy to actually achieve, because now those small interfering RNA is actually fully modified, not like a messenger RNA. So I think the conjugate is now the mainstream for the siRNA delivery. And I recently saw that some peptide RNA conjugate is actually investigated by an alum, but I'm not really sure their best candidate is out there yet. I think that answers the question. Yep.
Great. So for the last topic about immunotherapy for cancer treatment, how did you inject the particles?
So we did IM injections. We used model messenger RNA for expression of Ewha, and we made overexpressing cancer cells to challenge. So it was a basic model, and so far it worked pretty well.
Great. So intramuscular injection, right?
Yep.
Wonderful. Okay, I don't think we have any more questions. Then let's conclude this session. Let's thank our speakers again, Professor Lee, Professor Ming, and also Professor Hyukjin Lee. Now let me hand it over to Hyukjin.
Yep, thank you. Thank you very much. Thank you for all the speakers. I believe the symposium went very well and smooth, also very informative. The development strategy and the future aspect of lipid nanoparticle have been discussed well, I think. And I hope all the participants and audience enjoyed the talk, because it was very informative and also very attractive talks. Additional questions and answers will be further handled by the KAST. And finally, thank you again, all the speakers and organizing member of the symposium. Now we are going to close the symposium, and I'd like to say goodbye and keep safe. And for audience in Korea, enjoy your day. And participants from United States, good night. Thank you.
Thank you. Take care.